Bueno, buenos días. Buenos días. Estamos aquí reunidos el día de hoy para llevar a cabo nuestro tercer simposio de enfermedades por priones, un esfuerzo en conjunto de los integrantes de la Fundación FendMex, Fundación de Enfermedades Degenerativas México,
buenos días.
junto con los demás integrantes y ponentes que nos acompañan el día de hoy, donde tendremos una gran variedad de investigadores y clínicos que están precisamente haciendo estudios y el apoyo a las familias con ese tipo de enfermedades por priones. Esta reunión se realiza en función de que en el mes de noviembre es el Día Internacional de las Enfermedades por Priones y está enfocado a generar conciencia sobre estas enfermedades y a conocer algunas actualidades sobre el diagnóstico, tratamiento y prevención de las mismas. Agradecemos a todos los que puedan estar aquí con nosotros en este tercer simposio. Y sin más, le dejaré la voz al arquitecto Francisco Mota, quien es el presidente de la Fundación FendMex y al quien escucharemos para la sesión inaugural de este simposio. Muchas gracias. Arquitecto, ¿está listo para la presentación?
Arquitecto, creo que su micrófono está cerrado. No logramos escuchar.
Sí,
Listo.
ok, adelante.
Una disculpa, traía cerrado aquí el micrófono. Estimados invitados y ponentes, les deseo un muy buen día. Es un honor para mí darles la bienvenida a este tercer simposio virtual sobre enfermedades priónicas. Este día marca un nuevo hito para nuestra fundación, pues siempre hemos creído en el esfuerzo y la dedicación son fundamentales para construir proyectos que perduren en el tiempo. Nos complace compartir este momento con todos ustedes. Quiero agradecer a todos los que han trabajado arduamente para hacer posible este tercer simposio, organizadores hasta los voluntarios. Su dedicación y compromiso han sido fundamentales para la ejecución de esta jornada. Reconozco enormemente la extraordinaria labor de la Fundación Grupo México. Sin ellos, este simposio no habría sido posible. En este contexto, la Fundación FendMex organiza su tercer simposio sobre enfermedades priónicas, en el que participan ponentes nacionales e internacionales relacionados con el diagnóstico y tratamiento de las enfermedades priónicas, además de la investigación sobre posibles tratamientos. Este es un punto importante. Asimismo, presentamos las actividades de la Fundación FendMex y de la Red Internacional de Enfermedades Priónicas, Alianza Internacional de Apoyo del CJD, ISA, de Estados Unidos y de Australia. En este simposio aunamos fuerzas con el Centro Universitario de los Altos, que desarrolla proyectos centrados en el conocimiento de la fisiopatología de las enfermedades priónicas y cuyo investigador principal es el doctor Víctor Javier Sánchez González, fundador y director médico de nuestra fundación, a quien a su vez ha colaborado durante casi 17 años con la Red Internacional de Fundaciones de Enfermedades Priónicas, la Alianza Internacional de Apoyo a CJD y a su vez con varios centros de referencia y vigilancia epidemiológica de enfermedades priónicas en todo el mundo, principalmente en Estados Unidos y Escocia. Este tercer simposio sobre enfermedades priónicas se celebra este año, pero progresivamente ofreceremos la opción de contar de nuevo en nuestro país con principales exponentes en el abordaje clínico, el diagnóstico y el tratamiento de estas enfermedades, así como otros ponentes extraordinarios e investigadores, líderes en la materia. Este evento representa una oportunidad para reunirnos y celebrar juntos, fortalecer lazos y fomentar colaboración. Espero que este sea solo el tercero de muchos eventos que lo podamos encontrarnos y seguir trabajando por un futuro mejor para nuestra comunidad. Entre nuestros objetivos para este tercer simposio, nos proponemos, uno, difundir los conceptos básicos de los priones y las enfermedades priónicas. Dos, presentar y debatir los avances en el diagnóstico y tratamiento de las enfermedades priónicas. Tres, presentar nuestra fundación FendMex y exponer lo que hacemos. Cuatro, presentar la red internacional de fundaciones para las enfermedades priónicas, la Alianza Internacional de Apoyo CJD y su colaboración con FendMex. Les deseo a todos un muy buen día y juntos aprendamos un poco más de esta enfermedad. Nos presentan estos extraordinarios amigos y ponentes, los especialistas en la materia. Gracias por acompañarnos. Algo que de todos modos, digo, les quiero dejar ahí una nota muy especial. Es algo que se comenta, y lo comenté básicamente, y está en nuestra página web. La unión de las manos y los corazones de todas las personas hacen posible alcanzar los objetivos y de este modo ayudar a prevenir enfermedades, además de buscar incansablemente tratamientos o curas para las enfermedades neurodegenerativas. Muchísimas gracias. Y a continuación, como se indica en el programa, estará el doctor Víctor Javier Sánchez presentando los antecedentes históricos de las enfermedades priónicas. Muchas gracias, muy amables y bienvenidos.
Muchas gracias, arquitecto, por las palabras y por la inauguración formal de este evento. Estaré empezando a proyectar la presentación. A ver, déjame ver dónde está. Sí, sí pueden ver la presentación.
Sí.
Sí, ok, perfecto. Bueno, ya está, ya lo dejé en la pantalla completa, ¿sí se puede ver así?
Sí,
Perfecto, bueno, vamos a empezar entonces con el programa de nuestro congreso, o de nuestro simposio, y vamos a hablar un poco sobre los antecedentes históricos de las enfermedades por priones. Sabemos que, minimizo esto, em, que las enfermedades por priones han tenido bastante renombre, sobre todo a raíz de lo que se conoció como la epidemia de las vacas locas, que técnicamente el nombre es encefalopatía espongiforme bovina, y que fue una epidemia que ocurrió ya en el siglo pasado, hace ya casi unos 40 años, fue a mediados de los 80, pero que, como les comento, trajo a la luz este tipo de padecimientos y sobre todo de su agente causal, que es el prion. No, en su momento, sin duda, hubo una gran cantidad de información, como generalmente ocurre cuando estamos en primer contacto con una enfermedad, que se puede considerar como, en su momento, como emergente. Hay una especificación epidemiológica que se llama enfermedades exóticas emergentes y reemergentes. En su momento podemos haberlo considerado como una enfermedad emergente, no se había tenido una, una crisis, por decirlo así, de este tipo en el que muchos integrantes, pues, de ganados en Europa fallecían, pero eventualmente, alrededor de 12 a 15 años después, empezó también una serie de casos en los humanos. Se hizo la relación de los casos que presentaban una nueva variante de enfermedades por priones, que se conoció como la nueva variante, con la de la epidemia de las vacas locas o la encefalopatía espongiforme bovina. Bueno, a raíz de eso, tenemos que ver un poquito nada más algunos antecedentes de qué significa la neurodegeneración, que es el dato característico de estas enfermedades, y es un proceso de descomposición neuronal, mielínica y de degradación en general de tejido nervioso, y que tiene como características que su inicio es insidioso y que la evolución es progresiva. Prácticamente no podemos detener el avance de este daño y llevar a condiciones que pueden poner y que ponen en compromiso la vida de las personas, pero de lo cual hablarán más adelante algunos de nuestros ponentes. No me quiero meter mucho en eso. De manera general, todos los pacientes presentan demencia, un cuadro motor que es mioclonía, una especie de temblor fino, cambios en el electroencefalograma. El dato muy característico es que es rápidamente progresiva. Y aunque algunas variantes familiares pueden tener pulsos variables, algunos son mucho más lentos en su progresión clínica que otros, pero esos son los casos todavía más raros, ¿no? Son enfermedades, las enfermedades por priones, de una prevalencia muy baja. Entre las enfermedades neurogenerativas, sin duda la principal es la enfermedad de Alzheimer, y comparémoslo con la cantidad de casos que se esperan encontrar, si lo vemos por una tasa por cada 100.000 habitantes, pues es casi 1.500 veces más frecuente el Alzheimer que las enfermedades por priones, ¿verdad? Aquí tenemos este listado, pero sin duda tenemos muchos casos, ¿no? Se considera que hay alrededor de 1,5 casos por millón de habitantes al año en cada país, y eso lleva a, si lo estimamos en México, a que pudiera haber alrededor de 130 a casi 200 casos al año, lo que implicaría decir que cada día, un día sí y un día no, habría un caso por priones en el país. No, esto ya nos habla un poco de la magnitud del padecimiento, que parece que no es tan frecuente, pues casi cada día podemos encontrar un caso nuevo, ¿no? Bueno, hablando de las enfermedades, sabemos que hay de varios tipos. La más característica es la enfermedad de Creutzfeldt-Jakob. En muchos medios lo conocemos en manera muy global, las enfermedades por priones como CJD, por siglas en inglés, Creutzfeldt-Jakob Disease, y de ahí pues generalizamos con todas las demás variantes de las enfermedades, ¿no? Son enfermedades que se han encontrado tanto en los humanos como en otras especies, ovejas, en ganado vacuno, en venados y demás, y nos vamos a quedar también con este concepto. Son enfermedades que tienen el aspecto de que son esporádicas, hereditarias, iatrogénicas y que tienen un componente infeccioso, lo cual es un dato muy raro o atípico en la biología, en el que, por ejemplo, podamos heredar un agente infeccioso. Pero eso nos hablará más adelante en la siguiente presentación. Si nos damos a enfermedades por priones como tal, aquí tenemos un timeline que realizó el doctor Paul Graham, recientemente fallecido, y en paz descanse el doctor, uno de los líderes de la investigación de los priones a nivel mundial. Bueno, en esta imagen que él desarrolló, se puede observar cómo se tiene un primer registro de las enfermedades por priones desde 1720 en las ovejas, en registros en el Reino Unido, y conforme van pasando los siglos, ha habido hallazgos con otras especies. En 1920, prácticamente 2 siglos después del primer registro en ovejas, se tiene el registro del primer caso clínico de manera formal por los doctores Creutzfeldt y Jacob, y eventualmente ha habido una mayor cantidad de casos. Bueno, veamos cada uno a detalle. Ya lo comenté, de 1720 hasta 1932 se tienen registros no, muy intermitentes sobre lo que sería el scrape. El scrape sí se le conoce a la infección en las ovejas, y es una forma de describir ese rascado y actividad motora incrementada que tiene las ovejas cuando están infectadas. Que los registros no fueron propiamente por veterinarios, sino por los registros comerciales que realizaban los mercaderes, ¿no? Al hacer el intercambio de las ovejas, hacían las anotaciones de que la oveja estaba muy inquieta. Bueno, es una descripción que sin duda puede tomarse como una descripción clínica, lo que hace ver que este cuadro ya podemos registrarlo desde esta época. Una oveja con esas características la podemos ver así: es una oveja que empieza a tener una actividad motora muy errática, se empieza a rascar, empieza a ser un poquito más agresiva, y vemos cómo empieza a perder pues la lana, y además de tener autolesiones. Algunos años después se describieron casos de scrapie en ovejas en Alemania, y en el 98 más de un siglo, se empieza a reportar un estudio un poco más específico, más sistemático en los cerebros de las ovejas con scrapie, donde se reporta un dato muy característico de las enfermedades por priones, que es la vacuolización. Eso significa que hay agujeritos en el cerebro en este caso, de las ovejas afectadas, y esto le da también otro dato muy característico, que se le conoce como encefalopatías espongiformes. Da la apariencia que es como una esponja, un poquito agujereado el cerebro por este daño que está padeciendo, y lo podemos ver así, ¿no? Aquí tenemos estos huecos de los que les he comentado, que se pueden encontrar rodeando o dentro de las neuronas, ¿verdad? Estos puntos morados representan a las neuronas, a las células del cerebro. En este caso es un cerebro de una oveja, ¿no? Algunos años después se reporta la transmisibilidad del scrapie, lo cual es interesante porque ya empezamos a ver el dato de infectividad de estas enfermedades. Pero bueno, en 1920 dijimos que fueron prácticamente 2 siglos después, los doctores Hans Gerhard Kreuzfeld y Alfons Maria Jacob hacen la descripción en este artículo, ¿sí? Sobre una forma particular de enfermedades del sistema nervioso, el cual se publicó, ya lo comentamos, en 1920, hace la descripción de un paciente que había tenido problemas de memoria, cambios conductuales, una mala coordinación y trastornos visuales, y que posteriormente se, se categorizó como la enfermedad de Creutzfeldt-Jakob. Algunos años después, también por algunos investigadores alemanes, el doctor Joseph Gersmann, el doctor Ernest Streusler y Ilya Mark Schenker, describen la enfermedad que lleva su nombre, la enfermedad de Gersmann-Streusler-Schenker, y está la descripción del artículo. También hacen un título parecido sobre una enfermedad del sistema, una particular enfermedad del sistema nervioso, pero con carácter hereditario familiar. Es una de las características de la enfermedad de Gerstmann-Strauss-Schenker. Es una enfermedad hereditaria, es debido a una mutación específica, y que en el cuadro clínico que ellos describieron mencionan la ataxia, que es un problema motor, generalmente en la marcha, signos piramidales que pueden presentarse, por ejemplo, como rigidez muscular, y la demencia. Es una tocar muy característica de las enfermedades por priones, el que todos ellos cursan con demencia. En 50 y 60 se describe el puro, que también se conocía como negi-nagi, que es la forma de describirlo, que también era una manera en la que los habitantes de esta zona, es una tribu de caníbales en Nueva Guinea, hacían la descripción del cuadro clínico. Puro significaba también temblorina, y el primer investigador que tuvo contacto con este tipo de pacientes, Arthur Carey, utiliza ese término que le asigna a la población. Ese es como se le conoce ahora. Su primera descripción es 1951. Ahí podemos tener a Nueva Guinea, ¿no? En donde podemos encontrar a esta población, ¿verdad? En este sitio específico, Nueva Guinea, donde se encuentran estos habitantes. Ya no practican el canibalismo, es un canibalismo, o era un canibalismo ritual, es decir, consumían a su familiar o integrante de la tribu una vez que fallecía, en señal de respeto, y después se vio que por esta causa había propagación del agente infeccioso que es el prion. También hubo una descripción 52-53 por los antropólogos en la zona, Ronald Berry y Catherine Byrne. En el 53, John MacArthur realiza un reporte de la enfermedad, y se empieza a conocer un poco más este cuadro clínico, lo cual llama la atención de un par de investigadores, el doctor Gagdusek y Vincent Sigas, que reportan la enfermedad y le asignan una causa hereditaria, ¿verdad? Que no era propiamente hereditaria, sino que más bien era el hecho de que, como los familiares, en general los habitantes de la tribu se comían a la persona que había fallecido, pero solamente los familiares consumen el cerebro. Entonces, como se enfermaban solamente los familiares, pensaban que era una causa hereditaria. Después vieron el vínculo con el consumo del tejido cerebral. Hicieron un experimento muy interesante donde pudieron tomar muestras del tejido cerebral de personas que habían fallecido con el puro, lo inoculan en cerebros de primates humanizados, y observan que eventualmente desarrollan la enfermedad. Aquí se pudo tener una primera evidencia En general, en la historia de la medicina, de que la demencia puede ser infecciosa. Debido a estos hallazgos, el doctor Gagdusek obtiene un premio Nobel en medicina, por este tipo de hallazgos. También en el 59 el doctor Hadlock, que era un veterinario, al observar imágenes de los cerebros de las personas con Kuru, dice, es prácticamente muy parecido a lo que yo he visto en el Scrapie. Obviamente los cerebros de las ovejas con Scrapie, los hallazgos de pérdida de las neuronas, la vacuolización, que son los dos principales hallazgos, y se parece que puede ser el mismo agente. Esto genera esta pista, pero en su momento todavía no se pensaba en que podrían ser priones, se hablaba de que podrían ser virus lentos, ya que prácticamente cuando uno estaba en contacto con el agente infeccioso, desarrolla la enfermedad al menos 10 años después. Por eso por mucho tiempo los priones se les conoció también como virus lentos. Ustedes todavía buscan algunos libros de infectología, sobre todo los infectólogos son muy reticentes al concepto del prión. Para ellos siguen siendo virus, a pesar de todas las evidencias que ya hay. Pero bueno, en varios libros, todos estos los pueden encontrar los priones dentro del apartado de los virus, no como una entidad infecciosa por separado. Ya comenté el premio Nobel que recibe Gagdusek en el 76. Es en 1982 cuando, el doctor Stanley Prusiner, cuya foto vemos aquí en pantalla, obtiene el premio Nobel alrededor de unos 15 años después de que él acuña el término de prión. Después de una serie de experimentos, muchos de ellos muy elegantes para tratar de identificar si es un virus, una bacteria, o si es un virus, qué tipo de virus, y no encontrar absolutamente nada que lo lleve a virus, siempre encontraba una proteína, puede aislar a la proteína, aplica los postulados de Koch de la transmisibilidad, la replicación del agente y la generación de un cuadro clínico infeccioso en este caso, pues puede demostrar que hay un agente infeccioso nuevo, que es el prión. Él acuña este nombre, hace una fusión de las palabras infectious protein, proteína infecciosa, y la reacomoda para que tenga un nombre pues más o menos fácil de mencionar, que es el del prión, ¿no? Y también por este hallazgo, él obtiene otro premio Nobel de nuevo por la identificación de este agente infeccioso, cuya estructura química vemos acá a la derecha. En la parte de arriba podemos encontrar la estructura normal de la proteína. Es una proteína que cambia de conformación, tiene tres hélices como la ven en su conformación normal, y cuando cambia pierde una de las hélices. Estas hélices tienen una conformación distinta que la llamamos en hoja plegada, y eso es lo que genera la infectividad. Bueno, en la epidemia habíamos comentado que en 1986 ocurre una epidemia de las vacas locas, encefalopatía espongiforme bovina, bovine spongiform encephalopathy, que lo que significan estas siglas. Y aquí vemos el surgimiento de la epidemia. En el 86 era el primer caso identificado. A los pocos años se empieza a introducir una prohibición en el consumo de este tipo de alimentos, de los alimentos derivados de este ganado. A pesar de eso, vean cómo pasa un tiempo, al menos 5 años en los que llegamos a un pico y después se empieza a disminuir la cantidad de los casos afectados. También lo habíamos comentado, el mecanismo de transmisión, o más bien el periodo preclínico de la enfermedad, pues puede durar varios años, ¿no? Bueno, se reporta sobre todo en el Reino Unido, y eventualmente, lo veremos más adelante, se reportan los casos en los humanos. Pero también en esos mismos años se hace el reporte en la revista New England Journal of Medicine, una de las principales revistas en medicina, de este artículo que es el insomnio familiar fatal, y el título es Insomnio familiar fatal y disautonomías con la degeneración selectiva de los núcleos del tálamo. Y este artículo lo publican estos dos investigadores, Sergio Lugaresi y Pierluigi Gambetti, haciendo el estudio de una familia con el insomnio familiar fatal, una familia en Venecia, que durante varias generaciones había presentado muchos casos afectados con insomnio familiar fatal. Y el doctor Lugaresi se comunica con el doctor Pierluigi Gambetti, también han sido colegas cuando el doctor Gambetti estuvo en Italia, eventualmente ya estaba en Estados Unidos, y envía muestras de tejido cerebral. El doctor Gambetti empieza a estudiarlos de manera detenida, él es un neuropatólogo, y encuentra priones en el tálamo. En su momento, en el 86, habíamos comentado que prácticamente unos 4 años antes, cinco, el doctor Prusiner había empezado a identificar al prión. Solamente él contaba con los anticuerpos para poder saber si ahí había priones o no. Gambetti le pide los anticuerpos a Prusiner, Prusiner le manda una muestra, los inocula en el tejido cerebral y eventualmente encuentra una marca positiva y puede mostrar la evidencia de que esta enfermedad, el insomnio familiar fatal de carácter hereditario, pues es también debido a los priones. 10 años después, aquí vemos al doctor Robert Will, fue el encargado del sistema de vigilancia de enfermedades por priones en el Reino Unido y uno de los encargados del sistema de vigilancia de enfermedades por priones en Europa. Ya está jubilado. El doctor Robert Will en el 96 publica un artículo, lo vemos aquí con Ironside y colaboradores, pero ellos son los principales colaboradores en este trabajo, sobre lo que se conoce ahora como la nueva variante de la enfermedad de Creutzfeldt-Jakob. Generalmente para esta enfermedad le ponemos el CJD que comentamos, la V de nueva variante, y em, hacen la descripción de personas que desarrollan una enfermedad similar al Creutzfeldt-Jakob, una demencia rápidamente progresiva. Hay que comentar que es muy conocido que esas enfermedades principalmente se presentan en el adulto mayor por arriba de los 65 años, aunque sí hay casos que podemos considerar de inicio temprano, por debajo de los 65 años, y que son principalmente de carácter hereditario. Pero en este caso, las personas afectadas eran menores de 65 años. El cuadro clínico se presentaba alrededor de los 40 años y no era hereditario, entonces, pero era una demencia rápidamente progresiva. Entonces, había varias particularidades que los han pensado en enfermedad por priones, pero otras que no. Total que puede identificar a los casos clínicamente, pudieron hacer autopsias, aplican anticuerpos, encuentran al prión, y hacer una técnica que les permite identificar una variante específica del prión y correlacionarlo con los priones que se estaban presentando en los casos de la encefalopatía espongiforme bovina. Y de esta forma se obtuvo la correlación de que las personas se infectaban por consumir la carne contaminada del ganado que había desarrollado la encefalopatía espongiforme bovina. Em, También se conoce lo que es la enfermedad de Creutzfeldt-Jakob iatrogénica. Sabemos que iatrogénico significa pues, que es inducido por una causa médica. Y ha habido varios tipos de reportes, ya que estas causas iatrogénicas se han dado por trasplantes de córnea. Un reporte en el 74 nos habla de ello. Electrodos intracerebrales, como se reportó en el 77 por Bernoulli, el 74 por Duffy. En el 85 Koch reporta casos de enfermedades por priones por haber sido sujetos a tratamiento con hormona del crecimiento o por injertos dura madre, más o menos por las mismas fechas por Tadani. Hay que tomar en cuenta que en aquellos años estos tratamientos se realizaban de sujetos cadavéricos y pudo haber ocurrido, más bien pues este fue el caso, que las personas habían fallecido en la etapa preclínica, no habían desarrollado la enfermedad por priones, no había forma de sospechar en ella, pero en ese periodo de ventana de 10 años antes, ya hay suficiente siembra de priones como para poder producir infectividad, aunque todavía no desarrollen el cuadro clínico, y que fue lo que ocurrió en todos estos casos de trasplante de córnea, de electrodos, porque se intervino en ellos cuando ellos estaban en la etapa preclínica. No había forma de sospechar que tenían enfermedad por priones, pero ya tenían suficientes priones para producir infectividad. Posteriormente se observan los casos de transfusiones de sangre en estas personas. Sobre todo aquí se ha observado en las personas que habían o que eventualmente desarrollaron la enfermedad por tener la nueva variante, lo cual nos habla de la alta capacidad de infectividad y transmisibilidad que puede tener esta variante de los priones, que puede pasar y romper lo que se llama la barrera de las especies, es decir, de una especie pasa a otra especie distinta, del ganado pasa a los humanos, y de ahí de los humanos a otros humanos, ¿no? Em, y bueno, ya casi para concluir, podemos mencionar algunos hallazgos o datos que podemos tener de manera ya más de laboratorio, experimental, sobre las enfermedades por priones. Aquí comentamos este corrimiento en el tiempo, ¿no? Desde 1720 al 32, donde se hace este reporte de los casos de Scrapie en Escocia, lo que ya habíamos comentado, que se reporta la vaporización en el tejido cerebral, cuando se reporta la transmisibilidad del Scrapie y las similitudes con otros tejidos, bueno, con otros tejidos cerebrales de otros casos de enfermedades por priones en el 79, como también en el 82, Prusiner aísla a la gente que conocemos ahora como el prion, ¿no? Eventualmente se empezaron a desarrollar técnicas para elaborar casos transgénicos y sobre todo estos que podemos ver aquí. Nos empiezan a desarrollar nuevas técnicas diagnósticas, de las cuales nos hablará más a detalle la doctora Inga. El famoso caso de la amplificación cíclica de proteínas de mal plegamiento, el PMCA, que dio paso a lo que eventualmente se conoce como el RT-Quick, que es la técnica diagnóstica por excelencia. Creo que por ahí no. Em, y algunos otros hallazgos que tienen que ver con evidencias más experimentales sobre la estructura del prión, porque toda esa duda de cómo es que el prión, bajo un modelo que se llama de molde o de contacto, puede inducir el cambio de una proteína normal a una proteína normal, y eso es lo que se ha estado avanzando últimamente. Pero quizá el último hallazgo o avance ha sido este en cuanto al diagnóstico, y más adelante en el simposio estaremos escuchando a dos investigadores que están trabajando de manera muy ardua, exhaustiva y diríamos ya está exitosa para el desarrollo de dos nuevos medicamentos para las enfermedades por priones. Bueno, sin más, yo dejo aquí el micrófono para que si hay alguna duda o pregunta o comentario por parte de la audiencia, pues podamos responderlas en función de qué es la historia de las enfermedades por priones. Muchas gracias. Y bueno, creo que me toca a mí seguir comentando. No sé si hubiera alguna pregunta, duda o comentario por parte de la audiencia que podemos contestar en este momento. Si no es así, para eventualmente pasar a la siguiente plática que está programada para dentro de unos cuantos minutos. Así que podríamos aprovechar un pequeño, pues no receso, sino una pequeña pausa técnica para poder empezar a proyectar la siguiente presentación. Aquí estoy al tanto en los próximos minutos por si existe alguna pregunta al respecto. Gracias. Hello, Richard. How are you?
Oh, hi, Victor. I'm well. Are you?
I'm okay. I'm extremely happy to see you and hear you and know that you're going to be with us talking about the concept or the prion concept. I guess we have like 3 or 4 minutes free. I mean, I ended my presentation earlier than expected. I don't know if we can use this brief moment just to see if you can show your presentation. We have no
Yeah.
problem with that. Is that okay?
I will see if I can allow to share screen. I think that's share or a window. I think I want to share a window, don't I? Share this window. That's no good. Right.
There you go.
What am I sharing?
I guess we have your screen, your overall screen. I don't know if you can select your presentation.
I cannot see anything, strangely. Let me go back to share the screen then.
Okay.
Mhm. Right, that's here. So I need to, right, sorry, I need to find out what I'm sharing then. I don't know whether you can see that or not.
We can see your presentation. It says
Okay, right.
the Prion concept. I guess we're ready to go. Do you have any audio on your presentation?
No, I'm just talking, that's all. You
Okay.
I will talk along with it. Is that okay?
That's excellent for us. So I guess we're ready to go. Let me just do a brief presentation of Professor Richard Knight, which we have today with us in our third symposium of prion diseases. I will move to Spanish. Professor Knight, just to do
Yeah, sure.
the tutores in México. Bueno, tenemos con nosotros al profesor Richard Knight. La verdad me da mucho gusto tenerlo aquí. Es un investigador que ha colaborado de manera muy ardua con el profesor Bob Will, del que les hablé hace un momento, sobre la nueva variante de las enfermedades por priones. Y por mucho tiempo ellos, el doctor Richard Knight y Bob Will, han sido los encargados del sistema de vigilancia de enfermedades por priones del Reino Unido. El profesor Richard Knight fue el encargado, uno de los encargados de establecer el sistema de vigilancia en Escocia, posteriormente el Reino Unido, y posteriormente de establecer los sistemas de vigilancia de enfermedades por priones en Europa. Ha sido asesor de muchos de los centros de vigilancia de enfermedades por priones a nivel mundial, también de la Organización Mundial de la Salud, de la Cruz Roja Internacional, para establecer guías de diagnóstico y tratamiento de las enfermedades por priones. El profesor Richard Knight es neurólogo y profesor emérito ya en la Universidad de Edimburgo. La distinción de profesor en Europa, tendré que hacer este comentario porque algunos de estos ponentes son europeos, son profesores. Es una forma, es el máximo reconocimiento académico que se puede dar en Europa a un profesor, a un maestro investigador o al perteneciente de un claustro académico en una universidad. Es lo que puede ser el equivalente, por decirlo aquí, como profesor emérito. Y el doctor Richard Knight, precisamente profesor con ese tipo de distinción a nivel europeo. Em, y actualmente sigue colaborando con los centros de vigilancia. Es un ponente destacado en este campo. Ha colaborado de manera importante en la identificación de los casos de la nueva variante, de cualquier esporádico, de prácticamente cualquier tipo de enfermedades por pruebas que ustedes puedan conocer, a excepción pues, del CUR, obviamente, que sabemos que ya está prácticamente limitado. Y pues ha sido también ponente con nosotros en las 3 ediciones de nuestros simposios de enfermedades por priones. Colabora de manera muy activa con la red internacional del CJD International Support Alliance, que es uno de los integrantes, integrantes del Consejo Científico, y a su vez nos apoya a nosotros con cualquier cuestión que podamos necesitar desde el punto de vista científico o de colaboraciones con su país o a nivel europeo o con las fundaciones en las enfermedades por priones. Bueno, la verdad que hay bastante por hablar y por decir con el profesor Richard Knight, y sin más le cederé la voz para que podamos escuchar su presentación. I had to stop, Richard, because I know there's a lot to talk about you, but I just had to yeah, I know. I just had to stop so we can listen to your wonderful presentation. So the microphone is all yours, Richard.
Okay, well, thank you. Thank you very much for inviting me to talk. I was asked to give a talk by Victor on prion disease and the prion concept from the point of view of a non-technical person, a non-expert, to try to give background information to families and people affected not in a professional way. I'm sorry I have to speak English. So, prion disease, I think you've probably heard a little bit about this already, but there is a history of names here. Creutzfeldt-Jakob disease, an original name from 2 original writers. Then spongiform encephalopathy, a descriptive name, encephalopathy meaning disease of the brain, and spongiform because the brain takes on this spongy appearance. And then following transmissibility experiments showing that it could be transmissible, it was termed transmissible spongiform encephalopathy. But these days we tend to prefer the term prion disease, which reflects our current understanding. Now, there are prion diseases of animals like scrapie in sheep and goat, chronic wasting disease affecting cervid species, BSE in cattle transmitted onto other animals, and most recently a prion disease described in dromedaries. But I'm going to talk about the human diseases alone. So I'm going to give some background biology, which may be far too simple for some of you, but it's important, I think, that everyone has the understanding of the background, talk about the mechanism and causes of prion disease, and touch a little bit on issues of diagnosis and treatment, although I think some of these things will be covered elsewhere by others. So some background biology. I take it that everyone knows that the body is made of organs, like the lungs, the heart, and everything is made of chemical structure. Now, most cells are very small, but of course some cells are quite big. So a hen's egg is certainly a cell, it's very big, and things like the eggs that frogs and newts lay can certainly be seen by the naked eye. Whatever size they are, they all have the same structure. A surface membrane which seals the cell off from its environment, a central nucleus that contains important material, and in between a jelly fluid sort of stuff, cytoplasm, which contains also a lot of important machinery. If you open up the nucleus, it contains a lot of these linear structures called chromosomes. And if you open up the chromosomes, they contain things called genes. Now, genes, if you open them up, you find they're made of DNA. And DNA is a double helix structure of chemical units, which has been cartooned here. Now, DNA is a set of instructions for making proteins. So you might well say, well, why do we make proteins? Well, proteins have structural roles in the body. Collagen, for example, is a protein that's responsible for the tensile strength of skin and tendons in the body. But proteins also have functional roles. So a protein like hemoglobin carries oxygen around the blood. Now, if DNA is a set of codes for making proteins, you can look at it as a string of codes. Each individual code item is called a codon, and each individual item is responsible for bringing up one particular chemical structure, an amino acid. I've listed three of them, valine, methionine, and alanine here. They're relatively simple chemical compounds, and each codon, each unit of code in the DNA, is a code for one amino acid. So when the code is translated, you get a string of amino acids, and this string is the basic structure of a protein. Of course, the DNA is a double helix, as I've shown here, and this is because it contains two sets of the code. And so when you make a protein, first of all, there's transcription to form a single code, the RNA, and this is then translated to form the amino acid chain, the basic protein. However, once the basic protein chain is made, it's then folded in a complex way to make the final protein structure. This is a two-dimensional representation of it, but it's folded in 3 dimensions. The important thing is that the 3D shape of a protein governs its interaction with other chemical structures, and therefore it governs its functions. And you can see hemoglobin in a cartoon form in the left-hand bit of the slide here, showing a pretty complex shape, the kind of shape that's needed to carry oxygen around in the blood. Now, the shape of a protein affects how it behaves. And if you want a definition of life, at some level, life is how chemical structures interact with each other. And their shape affects how they interact and fit together. So you can see here on the left, this 3 triangles in red and blue is a certain kind of structure. And then another chemical unit comes along. you can see this arrow form, and it can interact with that structure because the shape is right. On the other hand, the shape on the right-hand bit of the screen can't interact with it. It's the wrong shape. Now, this is very important because interaction could mean all sorts of things. First of all, this interaction could be something that causes disease, or it could be something that prevents disease. So, for example, the arrow shape could in fact be a drug which interacts with the molecule on the left, which is some form of disease molecule, and leads to some sort of treatment. So, if we leave proteins in general and move to one protein in particular, the prion protein, a prion protein, usually designated by PRP, is one of our normal body proteins. And the responsible human gene, the gene that codes for it, is called PONP in humans. The functions of the prion protein are a bit uncertain. We know quite a bit about it, but its crucial functions are a little bit uncertain. The most important thing is it's a normal protein. All of us at this moment are making prion protein in our tissues, including our brains. So let's turn now to the disease, because what happens in prion disease is something goes wrong with the prion protein. So here you see the normal gene, PONP, producing the normal chemical structure of the normal prion protein, PRP. But then in disease, it changes shape and forms an abnormal shape. And it's the same chemical, the same set of amino acids, but it's now in a different three-dimensional shape. And this different three-dimensional shape will affect how it interacts with other molecules in the body, and so its function is changed. What is important in prion diseases, and this is indeed essentially the prion mechanism, is the abnormal protein, once it's there, is able to interact with normal proteins and turn those normal proteins into more abnormal protein. So once it's there, it tends to reproduce itself and produce more and more of the abnormal form. So this is the core prion concept. It's a self-replicating protein, a protein that reproduces itself without the need for genes or RNA or DNA. So this is the whole process in outline. The DNA of the prion protein gene is transcribed to form the RNA. This is then translated to form the normal prion protein, designated PRPC. Then something happens and it converts to the abnormal shape, which is usually designated PRPSC. Then the abnormal protein interacts with the normal protein to convert it, amplifying the amount present. But the abnormal shape does not behave in the normal way. It has different physicochemical properties, and it tends to aggregate and get deposited in tissue. And this kind of square at the top with this pinky brown stain is the abnormal clumps of abnormal protein deposited in the tissue of the brain in somebody with prion disease. And of course, why people get ill is because neurons, nerve cells in their brain, malfunction and die. And somehow or other, all of this protein pathology is toxic to proteins, causing them to misfunction and die. So why does this happen? Well, we recognize three causes: genetic, acquired, and idiopathic. So let's look at genetic first. Genetic in some ways is a bit easy to understand. A gene is a string of code, and well, mistakes could be made. Changes could occur in the string of code. And there are two important types of change: pathogenic mutations. These are mutations, changes in the gene code, which gives rise to a different protein structure, which is dysfunctional and causes disease. On the other hand, there are polymorphisms. These are changes in the code, but it doesn't make a lot of difference. The resulting protein still works and it doesn't cause disease. So let's look at pathogenic mutations. Basically here you have an abnormal gene. The code in the gene is altered, and I've therefore put a red star against it. It produces a slightly different form of the normal prion protein with a red star. And this is unstable in some way and more likely to misfold and then lead on to disease. So the abnormal gene gives rise to an abnormal protein and hence the disease. And once it starts, it then converts the normal protein into more abnormal protein. Originally, we talked about genetic CJD, Gerstmann-Strauss-Schenker syndrome, fatal familial insomnia, all names given to different varieties of genetic disease. But these days, most people prefer a different nomenclature, a modern nomenclature, and really talk about them just by the underlying mutations. So E200K, P102L are specific mutations, changes in the gene, which cause disease. What does that mean? Well, if you lay out the codon, individual code items on a table and just label them, number them from 1, 2, 3, 4, from left to right, then of course you have a whole string and each code is numbered. And at each code, the amino acid is specified, but if it's changed, then you get a different amino acid. So the number refers to the position, the number, codon number, the position in the code where the change has taken place. And the letters refer to the change in the amino acid. So E200K is a change at the 200th item of code in the prion gene, and the change is related to two amino acids, glutamic acid and lysine, which have code names E and K. Whenever you get a genetic prion disease, it's autosomally dominantly inherited, and that means that each time there's an interaction between someone carrying the mutation, here shown in the coloured square, with someone who doesn't have the mutation, the white square, each time they have an offspring, there is a 50-50 chance that that offspring will carry the abnormal gene. Now, I've shown here they've had 4 children. 2 of them got the gene and 2 of them didn't. But it's not necessarily the case. All 4 of them could have the gene, all 4 of them not, or 3 of them could have the gene and 2 not. It's just each time the child is born, there is a 50-50 chance that it carries the abnormal gene. Well, polymorphisms, the resultant protein is still functional. They're not directly disease causing. So this is just an example that may be useful. If you have a code for your holiday, the correct code instruction is your plane departs at 2100 hours. A polymorphism might be your plane leaves at 9 p.m. Saying 9 p.m. or 2100 hours are just different ways of saying the same thing. If you go at 2100 hours or at 9 p.m., you'll still get the plane. There's no dysfunction. A pathogenic mutation might be your plane leaves at 1900, and that, of course, is the wrong time and will lead to a mistake. Polymorphisms are not directly problem causing, but they sometimes incidentally make problems more likely. And if we just pursue this example, in my view, saying that the plane leaves at 9 is okay, but it's ambiguous because it could mean 9 a.m. or 9 p.m. And some people would say that using the 24-hour clock makes problems less likely. Well, the polymorphism that's relevant here mostly is something called M129V, and that means that at position 129 in the prion protein code, it may be coding for methionine or valine, 2 amino acids. And the normal UK population, about half of us are MV, about 11% are VV, and the rest are MM. All of us are normal, but this is just a variation that's found in the population. However, I mentioned that although it doesn't cause disease, it sometimes incidentally makes problems more likely, and I'll return to that a bit later. So let's go on to acquired disease. Well, this is in a way also fairly easy to understand in that abnormal protein from an external source goes into the person and causes the normal protein to change into the abnormal form. And then once that process has started, it forms a cascade which amplifies. Now, when you have an acquired disease, there's such a thing called the incubation period. The time from the exposure to when you develop symptoms of disease is called the incubation period. For most infections, like COVID, for example, or many viral infections, the incubation period is measured in days. But for prion disease, it can be very long, measured in years or even decades, and this makes attributing cause of acquired disease and preventing them and managing them quite difficult. There are three forms of acquired prion disease: Kuru, variant CJD, and iatrogenic CJD. Kuru is a disease of historical interest, limited to Papua New Guinea, and I'm not going to talk any more about it. Variant CJD is a human disease resulting from BSE contamination of food, BSE being a prion disease in cattle. In the UK, where it became a particular problem, it was recognised in cattle in about 1986, the first calves being infected in 1981. And prior to 1989, when certain food protection measures were put in place, nearly 500,000 BSE cattle entered the human food chain. However, although we were unfortunate in that some humans did get disease in the form of variant CJD, the number of people affected in the end was small, 178 in the UK. The last case was reported with onset in 2014. France was next, with their last case being reported in 2021, and other countries having just a handful of cases, and as far as I know, Mexico, none. There are now no known living cases in the world. Iatrogenic CJD, I think you've heard a little bit about, but basically dura mater obtained from cadavers and then used in surgery, and pituitary gland hormones obtained from pituitary glands and then used in medical treatments accounted for most of the cases. Neurosurgical procedures and corneal transplant accounted for very few cases indeed. If you look at iatrogenic cases related to treatment with cadaveric-derived human growth hormone, synthetic human growth hormone, which carries no risk of this, has been used since 1985. But as I mentioned earlier on, the incubation period for CJD can be very long, and in this instance, 5 to 40 odd years. So even though there's not been any exposure recently, we had 6 deaths in the UK between 2015 and 2023 related to this treatment. Idiopathic. Well, idiopathic strictly means of unknown cause. We also call idiopathic disease sporadic prion disease because it occurs sporadically throughout the world in every country where it's been looked for. We don't know the cause, hence the term idiopathic, of unknown cause. But the current feeling is fairly strongly that this is a spontaneous error. We make millions of molecules of this normal protein all the time, and as time goes by and we get older and older, mistakes might be made in the production of the protein, producing a slightly abnormal form. And the mechanisms in our cells which identify mistakes in protein production may become old and not so good. So as a result, at some stage in some people, there's a spontaneous error and a misfolded form arises which causes disease. It's the commonest form of human prion disease. Over 80% of prion disease is of this form. It results in 1 to 2 deaths per million per year in most countries where it's been studied. However, that is a rare disease, and it may give a false impression of how rare it is. If you instead ask what is the lifetime risk for anyone developing this disease, it's about 1 in 5,000. That's still actually fairly rare because certain diseases like cancer of the breast or cancer of the prostate have risk factors of 1 in 2 to 1 in 5. It appears to be just a spontaneous disease without any external cause, but there are variations in the clinical and pathological picture. Not everybody with this form of disease looks exactly the same. And this brings me back to polymorphisms. They're not disease causing, but they sometimes incidentally make problems more likely. And this M129V variation can affect the probability that you get CJD, even if it doesn't cause it. So this graph here shows the UK population, the normal population in green, and you can see, as I said, about half of us are MV and about 11% are VV. But when you look at the MM column, there are more people in the red with sporadic CJD than you'd expect by chance. And if you look at the MV column, there are fewer people with sporadic CJD than you'd expect by chance, showing that MM is in some way making people more likely to get sporadic CJD, and MV in some way making it less likely. In addition, it infects the disease in a way that when you get sporadic CJD, the clinical picture that results can vary with this polymorphism. And indeed, this has led to the idea of subtypes of sporadic CJD. You see differences in the clinical picture, the pathological picture, and diagnostic test results in different people. And this has been correlated with the 129 genotype and also the protein type that's found. There are two variations of the protein, 1 and 2. So people talk about MM1 sporadic CJD or VV2 sporadic CJD and so on. Well, I want to turn to diagnosis and make a few comments on this, although I think you'll hear more about this from others. This is a common question that we get asked by people. Why did it take my doctor so long to diagnose the CJD? I've looked about this, and actually the way that my relative presented was very typical for CJD. The real point is that the initial symptoms of prion disease are non-specific. What do I mean by that? I mean that these initial symptoms could be the initial symptoms of many other diseases, and some of these different possible diagnoses may be treatable. So the clinician has to spend some time thinking of these other possible causes and excluding them. So the initial investigations are done to explore other possible diagnoses, and then when CJD becomes more likely, things are done to support the diagnosis of CJD. These may be essentially non-specific. In other words, the abnormalities are helpful, but they're not confined to CJD, or they may be essentially specific, and that means detecting abnormal prion protein, which is the specific hallmark of the disease. If you look at clinical tests, the EEG, the spinal fluid 1433, and the MR scan appearances have been extremely useful, and they remain extremely useful, particularly the MRI, but they're not 100% specific. The abnormalities seen can be seen in other diseases. They're not specific in principle. There are two classic tests which are specific in principle: finding abnormal PRP in brain tissue, neuropathology, and of course finding a genetic mutation in the prion gene in someone who's clinically ill. But of course, in order to do specific neuropathology, you have to have brain tissue, which is either an autopsy and therefore after death, or a brain biopsy in life, which is rarely done. And the genetic test, of course, refers only to genetic forms of disease. What we do have is a new essentially specific test, the spinal fluid RT-QuIC, and I think you'll hear more about that. It's specific in principle and has therefore become very important in the diagnosis of sporadic CJD. I wanted to finish really with a bit of discussion on the possibility of treatment. We've moved, I think, from a false dawn in treatment to the beginnings of a real dawn. So if we are to think about how to treat prion disease, then rational treatment needs to follow understanding of the disease process. So if we remember that DNA is transcribed to RNA, translated to the normal protein, converted in disease to the abnormal protein, which then interacts with the normal protein and causes disease, and this results in toxicity to neurons, which malfunction and die. And these neurons, these brain cells, are responsible for everything we think and feel and do. So we could think about attacking this process at all sorts of places. We could reduce the normal prion protein or get rid of it, because if we reduce it, then there'll be no normal protein to be converted into abnormal protein. This is a perfectly reasonable step. Of course, it requires the idea that getting rid or reducing the normal protein doesn't have any ill effects because it is a normal protein. You could try to prevent conversion. You could try to get rid or clear the abnormal prion protein, and you could try to prevent toxicity, try to protect the neurons from the toxic effects. For various important reasons, which I don't have time to go into, most current development has been centering around reducing the normal prion protein. And it does seem, at least in principle, that one can reduce the normal prion protein in adults without necessarily producing any harm. And there are various approaches to this, and indeed several pharmaceutical firms which are now in the early stages of applying these treatments and researching them. The other methods, preventing conversion, clearing abnormal protein and preventing toxicity, have various problems or unknowns attached to them, but nonetheless they are areas of future interest. Of course, if you're going to treat somebody, the earlier the treatment you give, the better the outcome. But early treatment requires early diagnosis. And of course, it is the case that in a disease like sporadic CJD, by the time the diagnosis is made, the person is often very ill, very neurologically disabled. And it may be that the process, therefore, is very well advanced, and therefore treatment may not be all that effective. And I think we've learned a little bit about this from treatment of things like Alzheimer's disease, whereby, although we do have treatments that somehow affect the disease process, they're probably in general given too late. And that's one of the reasons that they don't have major effects. And symptoms may well begin some time after the disease process starts. So there is a very important question as to whether it's possible to diagnose early enough, even if we have got treatments that sound as though they're sensible. Well, prevention is the earliest form of treatment. And I think this is my personal opinion, that prevention in people who are carriers of a genetic mutation but have not yet become ill, that may well be the best hope that we have for treatment in prion disease, and the place where we're most likely to have the earliest and best effect. So I've been looking at this sort of stuff for over 40 years. And I think, when you're doing these things day by day, it really appears that nothing changes. And I'm just this quote from C.S. Lewis: day by day, nothing changes, but when you look back, everything is different. And it is different. Our understanding is far greater than it was. Our understanding of the prion disease process is now enormously better. Diagnosis is much better, and treatment for the first time shows real promise. So I think things are really a lot better than they were in the past. And with that, I'll finish, and I hope that I can return you to the original screen. I will stop sharing, and I am grateful to you for your attention.
Thank you so much, Richard, for this wonderful presentation. When we were once trying to see all of the topics that you're covering in the presentation, we're thinking like, will he have enough time? And you always manage to have a very, very wide explanation about prions, the history, some of the clinical treatments, in such a clear and understanding way that we're very, very grateful for all this effort that you give us and the families for the better understanding of the prions. So I have one question. Do you think we will ever have like an early or a biomarker for earlier stages of the disease? Because we usually get to think about this is CJD whenever it gets very rapidly progressive. Will there be any way of finding out before that?
Well, I think that's difficult. I mean, I think that we may well get biomarkers for the early diagnosis of the disease. Yeah, I think we may. But if you're talking about screening the population before they're ill, I think that's very difficult. Because of course it's a rare disease, and are you going to screen the whole population? But I mean, I think there's talks about screening for Alzheimer's disease in early middle life. And it may be that we would develop banks of screening tests that may be useful. I think that certainly is possible. In terms of clinical presentation, I've looked at this, and the thing that's really impressed me is we've got quicker and better at diagnosing CJD once people present to a neurologist. That's true. But of course, there are earlier phases. And when someone is a bit forgetful or a bit unsteady or feels a bit dizzy and they're 75, they don't rush off to a doctor necessarily. They or their family don't either. They think, well, maybe we'll wait and see because, these symptoms may be nothing at all and they may be very transient. And when I've looked at the time course of this disease and its presentation, those early phases, how long it takes for someone to think, oh yeah, there is something wrong, then to go and see a primary care physician, then get tested for very sensible things like, am I hypothyroid or something like that, all of that time, and then referral to a neurologist. By that time, something like, a quarter to a half of the disease process of sporadic CJD has already taken place.
Yes, I know that's very difficult to accomplish. I don't know, Francisco, ¿ibas a comentar algo?
Es solamente agradecerle mucho a Richard Knight su participación aquí con nosotros. Richard, we appreciate a lot your participation here with us. Thank you very much.
Thank you. Thank you.
Thank you very much, Richard, for that. We will be sending all of the speakers their letter of certification as a
Okay.
speaker
Thank you very much.
Awesome. We will move now to Dr. Michael Geschwin, who is right now with us. Hello, Michael. Are you ready?
Hi, yeah, let me just share my slides.
Okay.
Okay, give me 1 second.
Well, in the meantime, I will be presenting you in Spanish so that the audience can understand all of your or some of your accomplishments. I know they are very wide. So let me speak about Dr. Geschwind. Bueno, el doctor Michael Geschwind, quien ya se encuentra con nosotros, es profesor de neurología en el Centro del Envejecimiento y la Memoria en la Universidad de California en San Francisco, el que se conoce como Memory and Aging Center en UCSF, y se especializa en trastornos neurodegenerativos. El doctor Michael Geschwin es médico con un doctorado en neurociencias obtenido en el Colegio de Medicina Albert Einstein, y estudió en las escuelas de medicina de la UCLA, de la UCLA, posteriormente en la escuela de Johns Hopkins, en donde realizó estudios de neurología, y actualmente es el titular de la J. Homer Chair o la silla de neurología, precisamente en la Universidad de California en San Francisco. También es miembro del Consejo Directivo de Médicos de la comunidad, valga la redundancia, médica de San Francisco. Ha sido editor para la Academia Americana de Neurología en su edición de Continuum Dementia, y ha sido también titular del comité de la misma Academia Americana de Neurología para la realización de criterios diagnósticos en demencia, y ya tiene una amplia trayectoria en las enfermedades neurodegenerativas, sobre todo en enfermedades por priones, donde ha publicado una gran cantidad de artículos, tanto en enfermedades por priones como por enfermedad de Alzheimer, encefalitis autoinmune y de tipo límbico. Actualmente es un miembro muy activo en la comunidad de las enfermedades por priones. Es también miembro del Consejo de Asesores y Científico de las fundaciones de la enfermedad de cuello celíaco en Estados Unidos y la Alianza Internacional de Enfermedades por Priones, la CJD International Support Alliance. Y sin más, quisiera dejarle ya el micrófono al doctor Michael Geschwin para escuchar su presentación sobre un resumen clínico de las enfermedades por priones. I will now leave the microphone to you, Michael. We are ready to listen to your presentation. Thank you so much for being here with us.
I have a quick question. Maybe Victor, you know the answer. I realized I took slides from my last talk, and which was a Zoom recorded talk, and the slides are automatically advancing, and I don't want them to. Do you know how to turn that off by any chance? If you don't, I'll just have to go back and forth.
Let me ask our technician, Emanuel. Nos comenta el doctor Michael Gershwin que está utilizando la presentación que usó hace un tiempo y como estaba pregrabada a manera de video, está avanzando automáticamente. ¿Hay manera de desconectar esto?
Michael and Victor, you have to go in PowerPoint.
I have to do what?
If you go
Richard?
into PowerPoint, there's a whole section about slide advancement, and you just set it. So you'd have to go and edit it, and it should be edited so you don't automatically advance. There's a control in PowerPoint.
Yeah, I know. That's what I'm trying to find where that is.
It's in the top menu. If you look at the top menu, there's something about slide progression.
Maybe slideshow.
Yeah, in slideshow, things like that, and then it'd be slide sequencing or slide progression or slide changing or something.
Set up, hide slide, rehearse, record. Maybe I won't use timings. I just unclicked it. We'll see if it works. Okay, I'll give it my best shot, and if anybody knows, let me know. Okay, thanks, Richard. So my talk hopefully will follow nicely with Richard's wonderful talk. It's a little more technical, but I'm going to give sort of examples of the different prion diseases that Professor Knight just talked about. And I'll start with sporadic CJD, how it can be a classic and a non-classic type, and then acquired prion diseases, genetic prion diseases, and I'll talk briefly about some of our research findings that we're in the process of writing up for publication now. So as was already described, human prion diseases, they tend to occur in about 3 different ways: sporadic or spontaneous, or the majority, genetic, around 10 to 15%. One important point is that about half of a nice European study, it's about 20 years old now, but what they found is that of the people in the European consortia who they diagnosed with genetic prion disease by finding a mutation, 47%, almost 50%, had no known family history. So an absence of a family history doesn't exclude it being genetic. And that's why every patient we see, we test for genetic prion disease, because every once in a while, sometimes the mutation isn't 100% penetrant, which we mean if somebody has a mutation, a lot of mutations, everyone who has that mutation with almost 100% certainty, if they live a normal lifespan, will develop the disease. That would be 100% penetrant. Other mutations are less than 100%. Maybe only half the people with that mutation will ever develop the disease. And some mutations are as low as 10% penetrant, and others even less than that. And they're almost considered high-risk polymorphisms, which Professor Knight described. Historically, we've divided genetic prion disease into 3 subtypes based on their clinical presentation and their pathology, what they look like under the microscope when looking at the brain, into familial CJD caused by a few dozen mutations, GSS caused by about a dozen mutations, and then fatal familial insomnia. This FCJD almost always looks very much like sporadic CJD. Only really a true expert could differentiate it, and the family history would give the clue. But even pathology can be very similar to sporadic CJD and the clinical course. The GSS is a longer, slower disease, typically 3 or more years. It often looks like a cerebellar disorder, what we call a ataxic disorder, or a Parkinsonian disorder like Parkinson's disease, a little quicker than those disorders. And about 12 mutations cause this. Fatal familial insomnia caused by one mutation is responsible. And this is a disease that it's a little longer lived than sporadic CJD. Instead of being a half a year, it's maybe a year and a half survival. And they often have problems with the autonomic nervous system, the part of the nervous system that is automatic, that we don't think about, the nervous system components that control breathing, how our gut moves, things like that. Um, and then they get sleep problems. But you can see sleep problems in all form of CJD. They're just a little bit more severe in FFI. And then there are other types of mutations that aren't they weren't known. These types of prion disease, the first three I mentioned, they were named and described before we knew about the specific gene. And but since then, we found there are other types of mutations that present. Some can overlap. The octopeptides sometimes can look like FCJD or GSS. And then there's stop codon mutations. A stop codon is where the error in the gene, the mutation, actually makes a truncated, a shortened protein. It stops making the protein. So you get a small protein, and a small protein can actually lead to severe disease. And it can look very different than sporadic. One of our patients, he had onset in his late 20s and had the disease for over a decade. And we know that, and that mutation is probably not 100% penetrant because his parents didn't develop, his parent who had the same mutation didn't develop disease until 40 years later, 30 or 40 years later in their 70s. And then acquired prion disease, which Professor Knight mentioned, Carew, which I'll talk briefly about these. So this is the breakdown over about a 20-year period of patients referred to our center with rapidly progressive dementia, usually where CJD, prion disease, was very high in the referring doctor's differential. And we found that almost half of the patients didn't have CJD. They had another disorder. I'd say over the past few years, this percentage, the blue percentage, has decreased because our diagnostic tests for prion disease, the MRI and the RT-QuIC that Professor Knight mentioned, have gotten better. And Dr. Zaire will talk even more about these diagnostic tests and how they've improved our diagnosis. So for sporadic CJD, peak age of onset typically in the mid-60s. Youngest patient I'm aware of is 12 in Spain, and the oldest in Italy was, I think, 96. But typically mid-60s. If the patient's over their 70s, I'm a little suspicious of the diagnosis being CJD, though it doesn't rule it out. And although the genetic prion diseases tend to be younger, usually in their 50s, some of the mutations actually have older onset, even in the 80s. Mean survival, we're talking about a half year, but 85% live less than a year. In our cohort at UCSF, survival is closer to a year and a half. And I think that's, in a way, for two reasons. Number one, those are the more atypical cases, which we see more of. And number two, we spend a lot of time trying to find the first symptom of each patient, and the patients usually spend a few days with us. We see them multiple times, and whereas most of the literature is based on autopsy review, looking at medical records, we spend a lot of time trying to identify the very first symptom. And the reason is because by the time the diagnosis is made, it's too late, even if we had a cure. So I spend a lot of my research time trying to figure out how can we diagnose this earlier? What are the first symptoms we should be aware of and let the medical community know about so that they can begin to test for prion disease earlier? Because all the treatment trials are going to require patients to be very early in their disease. uniformly, on average, we find that um, that patients who come to us, that we find that their symptoms began on average about 6 months earlier than the medical records that even the family had initially stated. So that's why I think our patients have a longer survival, because we realize that the disease began earlier. There are about in Mexico, the incidence is about 1.2 per year on average in most countries. Switzerland is 1.9. There's a variability. Why that is. It could be because of their medical system. They have a uniform medical system, and they're probably it's a smaller country. It's probably easier to get diagnoses in certain countries and to get people into the medical system than a country like the US. In Mexico, we expect about 130 to 180 cases per year based on the population. But a person's lifetime risk, any of our risks of dying from CJD who don't have a familial CJD, is about 1 in 6,000. And that's data from the US CDC and from the UK as well. They came up with about the same number. So this is a classic case, a 61-year-old gentleman who had 3 months of problems with confusion, forgetfulness, problems with his vision, and problems finding the right word when he was speaking. He had trouble reading. He could read the words, see the words on the computer, but he told his wife he no longer understood what the words meant. He had trouble finding the right word when speaking. He had problems with his vision. He would look at his hands or look at another person, and their body parts seemed grossly enlarged. Their hands seemed large, their heads seemed large. That's a neurologic phenomenon called macropsia, and it's often in the back of the brain, either on the right more than the left, in the back of the brain here. And as we'll see on this patient's MRI, that's where we see the MRI abnormalities. We almost could predict where we're going to see the MRI abnormalities. He progressed. He had trouble doing chores. He could no longer ride a bicycle. And by the time we saw him, he had a lot of behavioral changes. He became frustrated, very upset with his symptoms, as you can imagine. He was very aware of his symptoms. Sometimes patients are not, but he was. This was his MRI. These are the two sequences that we require in the UCSF criteria. Other MRI criteria, like the European criteria, do not require these two sequences, but we've found that these two sequences are the most sensitive and specific, particularly when they're combined. So some criteria only require you to use this sequence called the diffusion weighted image, and then there's a complementary image called the ADC map. And what we're looking for is water molecules not moving appropriately through the tissue. They're being restricted. And when you have restriction on this sequence, the areas are abnormally bright. All the arrows are pointing to the abnormally bright areas. And on this sequence, some of these bright areas should be dark. And it's a little hard to see, but you can see here that it's darker here than here, and this is where the abnormalities are here. So you need to have some of the bright areas here, dark here. Though if it's late in the disease course, sometimes the brightness on this sequence will disappear, and you'll only see the dark areas. So we look at both the DWI and the ADC, but usually they're easier. The abnormalities are easier to see on this image. The other differences in our criteria are that not only do we require both of these sequences, we also allow frontal lobe involvement. And the reason why the European criteria didn't allow frontal lobe involvement is because in their studies, their radiologists called a lot of abnormalities as a lot of findings on the DWI as abnormal and consistent with CJD when the patient didn't have CJD. And that's because MRIs often cause artifact or abnormally bright areas in these regions. And our criteria show ways of removing that artifact. And that's why we allow the frontal lobe to be included, but you have to use certain sequences and techniques to remove artifact. Here's another case. This is a gentleman, a 66-year-old gentleman, similar age to the other one. But instead of having only 3 months of problems, this person had 2 years of progressive language problems. Our other patient also had language problems, but this person isolated language problems. And in neurology, we call isolated language problems primary progressive aphasia. Most of the time, this is a form of Alzheimer's disease. In this person, he had trouble naming. He had trouble understanding long sentences. He still could speak very fluently. When you spoke to him, it seemed normal. Occasionally, though, he would trip up on a word or wouldn't understand a word that he should have understood. He was very active. He was still mountain biking, kayaking, doing yoga, and driving a truck 2 years into his diagnosis. We followed him we diagnosed him with CJD based on his MRI. Here's the DWI, and you can see again these abnormally bright areas here. And this is the left side of the brain. The images are reversed here. Left is right, right is left, and all the images I'll show you. And this is where language, a large part of language, is processed in the brain. And we could have predicted this area would be involved based on his language symptoms, his aphasia. And so we diagnosed him with CJD, and he ended up dying from path-proven CJD after more than a year later, about 3.5 years into his course. And we published this paper. So one of the messages I'm trying to get across here is that where the abnormalities are occurring in the brain, where the abnormal misfolded protein is accumulating and causing nerve cell damage, that determines the symptoms that we'll see. So if the occipital lobe and back here is involved, and we'll often see MRI changes in that area, then we'll expect visual symptoms. If the parietal areas over here are involved, we might have problems not with vision, but are also processing of vision if it's on the right side of the brain. If it's on the left side of the brain, we might have trouble with some language, with grammar, with the ability to do learned movements. If the limbic system, which is shown here in blue, and this part here is involved, we might have problems with memory. If the frontal lobe is involved, or the connections to the frontal lobe, behavior and personality. If the deep nuclei, which are shown here, are involved in the center of the brain, movement problems, cognitive problems with connections to the frontal lobe, change in personality. If the thalamus is involved, which is also here, the thalamus is sort of the central connector to many regions of the brain. You can get pain syndromes, problems with the autonomic nervous system, sleep problems, and many, many other problems. So sometimes I call CJD or prion disease the great mimicker, because depending upon what parts of the brain are affected, it can look like almost any other neurodegenerative disease, Alzheimer's disease, Parkinson's disease, even Huntington's disease. So we've shown at our center that there are 3 main patterns on MRI based on our cohort. The surface of the brain, or the cortex, can be involved, or the deep nuclei shown here. This is the caudate. And we find that in our cohort, about 2/3 of the patients have this pattern. About 1/3 of patients have predominantly only the cortex involved, with little to no involvement of the deep nuclei. And then about 2 to 5% have predominantly deep nuclei involvement. And the pattern of MRI can actually help us subtype the form of CJD. And Professor Knight talked about the codon 129 polymorphism. We can generally, to some extent, predict the codon 129 polymorphism. So for example, in this case, this patient likely has VV2 or MV2 subtype of prion disease, where this patient probably has MM2 cortical subtype, and this patient probably has MM1 or MV1 subtype. It's not perfect, but it generally can help us. we found that these MRI abnormalities have very high sensitivity and specificity um, when read properly. But uh, our center and in the UK, in London, they did a beautiful study where they found that only half of their MRIs were read properly by radiologists, meaning a patient had CJD, the MRI showed the abnormalities, but the radiology report didn't note that CJD should be considered. At our center, it was even worse. Only 36% of cases referred to us from mostly from around the US did the MRI report mention CJD. And of the ones that were missed, so that means 66% were missed. It turns out that about two-thirds of those, or maybe it was about half of them, they noticed the abnormalities like these, but they didn't know to attribute it to CJD. And then in the remaining half, they didn't even notice the abnormalities. They probably thought it was artifact. So you need somebody who really knows how to read the MRI. And sometimes neurologists can be best at that. But sometimes radiologists can be good too. Acquired human prion disease that Professor Knight referred to, Kuru, I'll talk briefly about iatrogenic. So this was due to a practice of endocannibalism in the Faray linguistic group in Papua New Guinea, where they they used to honor their deceased relatives by eating them in a mortuary feast. And it was the women and children who tended to eat the parts of the body, the brain, the spinal cord, that had higher concentration of prions, and they tended to be the ones that got the disease. And then when that practice was stopped, the disease gradually died out. Though we know that the incubation period, meaning the time from exposure to getting the disease, can be as long as 50 years, also work from the United Kingdom. And then iatrogenic means medically induced. Basically, a medical accident caused or a mistake caused the transmission of the disease to a patient who is undergoing a medical procedure. And then variant CJD from exposure to bovine spongiform encephalopathy or the prions from that. So Kuru, I've already told you a little bit about it. At peak, 1 to 2% of the population was involved, women more than men for the reasons I described. The men had certain parts of the mortuary feast, and the younger children were affected as well as the older as well. And then this is, I think, Vigas and Carlton Gajasek and Vigas from Australia examining an unfortunate young boy with Kuru. So for iatrogenic CJD, just these are some numbers. It's about this is 13 years old now data, but there different surgical procedures have led to accidental transmission, and medical procedures have caused it. And there have been 3 to 4 cases of blood cells that primarily in the UK, in which people who were exposed to bovine spongiform encephalopathy and later developed variant CJD, before they became symptomatic, their blood already had infectious prions in it. They donated blood, not knowing that they were at risk for transmitting the disease. The recipients of that blood ended up years later developing variant CJD. This is the only known prion disease that I'm aware of that has really conclusively been shown to be transmissible by blood or blood products. It's theoretical it can be transmitted by other diseases, but the data for that is lacking. Um, um, we've seen 2 or 3 cases at our center from people in the United States. All the people we've seen acquired their disease outside the United States and then moved or came to the United States for a visit. One woman we saw was 20 years old. She was in the British Communication Corps, basically very high level, very intelligent young woman. In September of 2001, started to become depressed, changed in personality, less social. She was actually given a psychiatric discharge from the military. They thought maybe she had schizophrenia or psychosis because of her age. That this is the peak age of onset for psychosis for many women. Then later she developed other symptoms, neurologic symptoms that made them realize this is not schizophrenia or psychiatric disorder alone. This is a neurologic disorder. And they diagnosed her with variant CJD. She came to, excuse me, our Center for Treatment Trial, and unfortunately, eventually passed from the disease despite being on our treatment. Her findings on MRI, she had something called the pulvinar sign, which the radiologists in the UK identified. And it's where the posterior, this is the thalamus, and the posterior part of the thalamus, which is called the pulvinar, is brighter than this part of the brain called the putamen. And that's the pulvinar sign. And it has a relative degree of, medium sensitivity, meaning a majority of patients with variant CJD will have this, but many do not have this sign. And then in terms of the specificity, it's relatively specific for variant CJD, but there are cases of sporadic CJD that can have the identical sign. So it's a strong indicator that it could be variant CJD, but we wouldn't make a diagnosis of variant versus other forms. And then some other diseases not related to prion disease, like metabolic disorders, like deficiency of certain vitamins, can sometimes also cause similar findings. And yeah. And then so patients with variant CJD, they tend to be younger. So instead of patients being in their 60s, they're usually in their late 20s on average. The age range is 12 to 74, but vast majority are less than 55. Their survival is longer, about a year longer than sporadic CJD. The EEG is different. The MRI tends to be different. They have different genetic susceptibility, that codon 129. Almost the vast majority of patients all had codon 129 as MM, though there have been rare cases who were MV. And the distribution of the prions is outside not only in the nervous system, but also in the lymphoreticular system, the lymphoid tissue lining the gut and whatnot, and in the adenoids and in the back of the throat. So that can be another way to diagnose the disease. And we tend not to see that with the other diseases. No cases to my knowledge in Mexico, 4 in the US, 2 in Canada. None of them were acquired in North America, though. And in 21, Professor Knight referred to the, I think last cases, I think was 2021, and they were actually accidental transmission from a mouse, a humanized mouse, a mouse that had the human prion gene, was infected with variant CJD, and the lab worker wasn't careful, and they stuck themselves with a needle from working with that mice. And then a few years later, they came down with the disease. A real preventable tragedy. I think I'm going to skip these because Professor Knight really talked about the relationship between BSE and variant CJD, and the last cases being in 2021. And then I'll talk about the prion protein gene. So this is a schematic of the prion protein gene. It's about 230 plus amino acids. This is the protein, and it tends to be cleaved right around here. In red are the mutations. In blue are some of the novel mutations we've identified at our center. And actually, I realize we're missing some. There are some that aren't here. But these are just a few. And then below are polymorphisms that Professor Knight referred to that can affect one's ability to get the disease or might modify how it presents, but they probably don't, in most cases, they don't cause the disease. The exception being this V180I, it probably does cause the disease, but at a pretty low rate. It only has 10% penetrance. And here's that polymorphism, the most important polymorphism that affects one's susceptibility for getting the disease, and it can alter how the disease presents. And then this is a variant that a group in London at the MRC showed that this genetic polymorphism can protect one against variant CJD. This is a beautiful study that was done. And actually, I should have cited it here, but I realized I don't have the citation down below. So here's another model of schematic of the gene. And in red are the mutations that tend to present as F familial CJD, or what I call here genetic JCD. The green mutations are the ones that present like that slower form of the disease, Gerstmann, Strauss, or Scheinker. The blue is the only mutation that presents as FFI. And then the nonsense mutations, this is where the protein is truncated. It stops being made right here. And this is the novel mutation, one of the ones that we identified. And this is one identified in the UK. And I can't remember where these other ones were identified. But these tend to present very different. This patient was thought to have a disease called frontotemporal dementia and was actually in our frontotemporal dementia research project until our genetic counselor for prion disease happened to see the patient and said, I wonder if this could be prion disease. And we did the genetic testing, and sure enough. And then not all of these mutations are 100% penetrant. Some of them are like the E200K, which is the most common mutation in the US, Mexico, and the world. This one right here. This has somewhere between 60 to 90% penetrant, meaning between 10 to 40% of patients who have the mutation will never develop the disease. Why that variation? Well, in Slovakia, where there's a founder, they reported 60% penetrance. In Israel, where there's a founder from Spain and Portugal, a Spanish Jew, or what we call Sephardic Jew, whom they were, many of them were kicked out of Spain during the Inquisition, and they moved to many moved to North Africa. Eventually, many of them moved to Latin America. And some of them, all of them were Jewish, but some of them converted to Catholicism in order to save their lives and not be killed or be forced out of Spain during the Inquisition. And so now the families in Latin America are Catholic, but almost all of them we can trace back Jewish ancestry. And in some of them we can identify because unknowingly the family still practices Jewish practices. A classic example that I hear quite frequently from these families is that the grandparents would light candles on Friday night, which is a Jewish custom, but not a Catholic custom. And they often would do it outside, away from windows, and that's because they didn't want other people to know that they had Jewish ancestry. So they would light the candles away from windows, sometimes even in the basement. So it's the most common mutation worldwide. There are at least 4 geographical origins to this mutation. I already talked about the 2 most common, but there's also Japan, and then there's Austria, Hungary, Germany, Italy. And actually, I'm not sure how separate these are from each other, or whether it's all a common founder. And others might know the answer to this better than I do. And they often look like sporadic CJD clinically if you didn't know the family history. And this is okay. And then these are These are the three gentlemen who first described GSS, um, um, and from Russia and from Austria. And this is, I'm going to present a family of ours. The daughter presented at the age of 23. She had a troubled past. She did a lot of drugs, and in March of 2003, when she was 23, she started to develop these progressive symptoms. So some motor symptoms, increased thirst, depression, problems with her gait. Then by 2 to 3 years in, she developed more cognitive problems, and which just gradually worsened over time. And then her father developed his symptoms, even though he's 26 years older than her. He developed symptoms the exact same month, but his were slightly different order. He started with personality changes and only had cognitive changes a little bit later than motor, more behavior, and but they both carried the same mutation. And some of the tests that we typically do for prion disease at the time, like the CSF 1433 was negative. Their MRIs did not show that any of the abnormalities I showed you. What they showed was atrophy or volume loss, which isn't specific for prion disease, but we see it in typically in GSS, especially patients who have a long time course. Okay, looks like I'm getting a little over time. This is their family tree. So this is the daughter. This is her father, her father's brother, her uncle, and then you can see the grandparents' generation, the great-grandparents, and the great-great-grandparents. They're all shown in red, as we presume that they had GSS based on their early death and their symptoms. The gene test wasn't available to above this generation. Um, a lot of the data I present to you is because we see patients over a 2-day visit historically at our center. We do a lot of different testing for them. We do cognitive testing, we take blood, spinal fluid, we do the MRI. They see a genetics counselor, social worker. We do a lot of scales with them. We do a clinical meeting afterward, and we see patients who have symptoms and patients who don't have symptoms, who are at risk for genetic prion disease, who may or may not have the mutation. Um, and we have a model for what we predict. We divide our mutations into the familial CJD type, which are fast. These look like sporadic CJD. And the slow, these would include the GSS and the stop codon mutations. And we think that each of them will have a different trajectory. If this is their onset of clinical symptoms, our prediction is that before onset, we're going to begin to detect abnormalities. And we have found already we and now at least four other centers in the world, in the UK, Israel, and in Boston, have found that our E200K mutation carriers, some of these people will develop a positive RT quick. And to our knowledge, that RT quick can be positive as far as early as 4 or more years, 4 to 5 years prior to onset of symptoms. Um, so these are people that when we do have a treatment, we're going to want to be following their spinal fluid and doing RT quick. And when we see that increase, that's certainly going to be a time we might want to consider treatment. Our centers also found MRI abnormalities. This is some we can find in individuals. It's an RT quick as we can do in individuals. In the MRI, our data shows if we take all of our E200K patients or all of our fast carriers, as a group, we're finding reduced volume and reduced what we call mean diffusivity um, on MRI. We don't see it by eye, so you don't see those abnormalities those bright abnormalities on the DWI and dark on ADC. We don't see that by visual assessment, but when we actually use computers to analyze the and quantify the diffusion, it's abnormal, but not enough to be detected yet by the human eye. And for the GSS, unfortunately, our cohort we think is too small to have made. We haven't yet identified anything really in the early phase. We do know that before symptoms become obvious, there's brain volume loss. Um, yeah. And yeah, this just shows some of the people we've seen in our study from genetic families, and we have far more fast than slow carriers. And I think I'll skip this, but these are the most common we see is the E200K, followed by the P102L, D178N. So this one is a fast mutation, a fast, slow, fast, slow, slow, slow. So, uh, and then in blue are people who are carriers, and then we see their unaffected family members as controls because they're a good control because they share all other genes, many other genes similar, except the non-carriers don't have the mutation in the prion gene. So just to end, human prion diseases are very diverse. Diagnosis can be difficult, but our methods are improving, and Professor Zare will talk about that. We can learn a lot about sporadic, the more common sporadic disease, by studying genetic prion disease. And I didn't talk about it, but this idea that Professor Knight talked about a prion forming, a misfolded protein causing other proteins to misform, we now know that to be a common mechanism of other neurodegenerative diseases like Alzheimer's and Parkinson's and multiple system atrophy and probably Huntington's disease. So some people actually call all these diseases prion disease. Other people say, well, they're prion-like. But regardless, this idea of a protein misfolding and causing other proteins to misfold, we now know is not unique to PRP prion disease. And I think I'll end there. I want to thank all the work I've shown you. The research is very collaborative, both colleagues at UCSF and then colleagues from around the world in the US. And I want to thank the US National Prion Disease Pathology Surveillance Center. They do autopsies on patients, and it's paid for by the US CDC. And they allow us to do prion typing and provide a great service for us that's funded by US Congress to the CDC, and we hope that continues. And this is a picture of our center. Thank you very much.
Thank you so much, Michael, for this great presentation on the clinical aspects of prion diseases. I find that study on the preclinical biomarkers on prion diseases very interesting. See? Oh, sorry. So what do you think could be a good biomarker for the preclinical stages of CJD, if there gets to be one?
Well, I think right now the best biomarker for at least the E200K is the spinal fluid RT-QuIC. That is pretty clear. We're in the process of having our patient's blood and spinal fluid analyzed, looking at several hundred other biomarkers to see if any of them are as good as or even better than the RT-QuIC. We don't know how sensitive the RT-QuIC is in E200K for people who are pre-symptomatic. All we know is that from looking at all the data from at least 4 centers in the world and probably more now, to our knowledge, everyone who has developed a positive RT-QuIC eventually has developed symptoms, and that's been within 5 years or less. So we don't know if that will hold up, because it's still a very small number of people. So we don't know if everyone who is at risk for developing the disease will have a positive RT quick and when. But that's why we need to continue doing the research. We need patients to volunteer to come in. We need to be able to take blood, and particularly spinal fluid. We need to look at the brain by MRI. Unfortunately, my funding ran out for this study, so I'm applying for more funding. But I don't know if it'll happen. It's a scary time, as you know, in the United States for research. We'll see what happens. But research is certainly going on at other places around the world.
Okay, thanks. I don't know if Francisco wants to say something up.
It's only to make thanks to Michael for being with us. Michael, thank you very, very much with being with us here in this symposium. We appreciate a lot your collaboration here.
Great. Well, thank you, and thanks again for having me this year. It's always a pleasure to speak to this symposium.
Thank you so much, Michael. We also thank Inga and Dr. Ryan Napoli for being here already for this symposium. Just one note. I wrote in the chat the link for the next block of the transmission. We had to do 2 blocks of the transmission because of the licensing of Zoom. So we will have to close this one, but we're opening the one that I already put on the chat. So if you want to join us, and obviously you will have to join us because you're both the speakers of the symposium. Maybe you want to click that one. That will take you to the other session that we're opening in 1 or 2 minutes. Is that okay?
So, Victor, just to ask, so this will be also for us, for the speakers, right?
Yes.
Okay.
This is only for the speakers because we're broadcasting in. Yeah. Okay. So you can try that, and it will take you to that session.
Okay. Yeah.
Oral surgery. So I have to look now for emails that I missed, and I want to catch up on your genetic prion disease.
Yes, please. We plan a meeting in Vienna next year. So maybe this is might be an interesting destination for you.
Yeah.
So
Vienna's one of
Vienna
my
in
favorite
September.
cities. Do you know when it'll be yet?
It will be someone between 13 and 14 of September.
Oh, okay. Great. I'll put it on my calendar.
Okay, so we're moving to the other session. Do you already have the link?
I'm now, I think, in the other session.
Okay. Okay, Michael, you're all obviously welcome to meet us with the rest of the speaker to the other link.
Oh, I have 2 links open. Oh,
Yeah.
so let me close one.
Yeah. So
Yeah, I'm probably going to sign off because I have to get to clinic. But thanks. Great to see you.
Great to see you, Michael. See you
And
soon.
thank Richard for helping me with that Zoom issue.
We will.
Okay.
Thank you so much.
Great.
See you soon.
Bye.
Francisco, ¿están conectados en la otra sesión?
No, todavía no. Estoy tratando de ver cómo me conecto, ¿no? O sea, nada más me enviaste una, ¿no?
Creo que en el chat.
En el chat ya está, ¿verdad?
Victor.
It's
El
not working.
mismo, ¿eh?
It may not be working because we have this one opened.
Okay.
So we will need to close this one. I just want you, maybe you can copy the link. Is that okay? Hope that works. If not,
I will try.
okay. Please copy the link that we have on that chat.
Okay.
Our,
So just in case I disappear, you know I'm trying.
yes, I will send you the link
Yeah.
again
Okay.
by email. Let me email it to you. I will close this session right now.
We know. Let me see. Okay, here is Victor already.
Okay,
He
so
is
we got,
already there. Okay, perfect.
now if we got Brian, he was connected. Let me just email him the link again. And Brian,
Okay, here is Victor already.
yes,
He is already there. Perfect.
Salió en-
Brian,
no,
he was connected.
apenas, pero no hay video.
Email him the link.
Ya,
again.
ahora sí está saliendo. Justo
And,
ahorita.
so,
Victor already.
nada más para la parte técnica, creo que se está transmitiendo en este momento lo que estamos platicando por el YouTube. No sé si pudieran hacer una pequeña pausa. We're almost there, Inga. First of all,
Okay.
thank you so much for yeah. So we're working on having the Arctic Week finally in Mexico. So hopefully during next year we will have that as for the diagnosis of prions, obviously. And
Perfect.
we'll be very, very, very happy to let everybody know that we are now, we will be a lab on having the Arctic Week for the diagnosis of prion. This could be made.
We are already.
Okay, so we've been told that we're ready for the transmission. I guess we're ready, and I will present you Inga
Okay.
in Spanish because that's Dolores is in Mexico, and then I will let you speak about the diagnosis of prion diseases. Bueno, estamos el día de hoy continuando con nuestro simposio de enfermedades por Priones. Tenemos que hacer un pequeño corte técnico para brincar de una sesión a otra, pero estamos de regreso aquí el día de hoy. Se encuentra con nosotros la doctora Inga Ser, que es neuróloga, la doctora Inga Ser, que a su vez es profesora en la Universidad de Göttingen. Luego lo podemos ver allá arriba en nuestra presentación, y a su vez es encargada del Centro de Referencia en Alemania para las enfermedades neurodegenerativas, en donde tienen a su vez la división del Centro de Referencia Nacional para Enfermedades por Priones en la Universidad Josh Augusta en Göttingen. La doctora Inga Ser, como ya les he comentado, es neuróloga, atiende a pacientes en el hospital, y a su vez es la encargada del sistema de vigilancia y es la principal promotora de establecer los criterios diagnósticos para enfermedades por priones. Como ya les comenté, la distinción de profesor en Europa es una de las más elevadas o la más elevada distinción, y les había comentado que varios de nuestros ponentes tienen este nombramiento. Así que la doctora y profesora Inga Ser estará con nosotros hablando el día de hoy sobre estas estrategias de diagnóstico. Y sin más, quisiera dejar el micrófono para escuchar su presentación, y le agradecemos de nuevo el que se encuentre con nosotros. We will now pass the microphone to you, Inga, and we're ready to hear you. Thank you so much for being here with us today.
Yeah, thank you very much. I'm really happy to be here with you, and also because we have this long-standing friendship and collaboration. So, of course, there is a very important axis, Germany and Mexico, so from my point of view. Yeah, so thank you very much for inviting me to talk about the diagnosis in prion diseases. And what I would like to tell you is something about the progress which has been made in this area in the last years or decades. So starting with the first systematic studies in the 70s, there was only the brain biopsy or autopsy which allowed the definite diagnosis of prion disease. And of course, this was a very invasive and dangerous situation for the patients. Later on in the 80s, there was the electroencephalogram, which was used to support the diagnosis because, of course, the clinicians, we did not have any other options. But later on, when we started to do all these systematic studies on prion diseases, when the surveillance centers were established throughout Europe and the world, there was I would say, important interest on developing this area. And for example, cerebrospinal fluid, magnetic resonance imaging, and nowadays blood are already on the way to support the diagnosis. So there was a very important development in the area. Michael Gershwin told you about the magnetic resonance imaging. Therefore, I will not spend so much time on it, just to confirm that this is how we see the situation. There are very typical changes in the magnetic resonance imaging, which are very typical and support the clinical diagnosis. We've always been interested how the MRI looks like in sporadic Creutzfeldt-Jakob disease, and then we identified several patterns of these MRI changes. For example, there are some patients which just see high signal in the cortex and in the basal ganglia, but then there are patients that have just cortical changes, and then there is another group, they have only changes, symmetrical changes in the basal ganglia and in the thalamus, but no cortical changes. And even 7% of all patients have a normal magnetic resonance imaging. But this this pattern of different MRI high signal changes is very intriguing. And of course, we are always try to understand why some patients develop this type of changes, the others have other picture. And there was an attempt also if this might be related to the molecular subtypes of Creutzfeldt-Jakob disease, for example, those with a very frequent subtype like MM1 and MV2, they have a typical characteristic asymmetrical changes in the cortex and in the basal ganglia. And the others, as Michael already told, for example, the symmetrical changes in the caudate nucleus and thalamus, they are more likely to belong to the subtype VV2. So there is, I just repeat what he said. I would like to focus a little bit more on the cerebrospinal fluid tests in Creutzfeldt-Jakob disease. And the first thing what we always need to consider is that the routine cerebrospinal fluid tests in CJD are normal. So there is no change in the routine tests, like there is no sign of an inflammation in this cerebrospinal fluid. Also, we know the disease is transmissible, but it does not cause a typical inflammatory response. So we don't see elevated cell count. We don't see abnormal albumin ratio. We do not see usually high protein, and also the ratios for IgG, for immunoglobulins IgG, IgA, and IgM are normal. So this is, it appears from the first test results which we get as neurologists or as clinicians that there is nothing very abnormal in the cerebrospinal fluid. Nevertheless, there was a progress in the biomarker field because the cerebrospinal fluid is a platform where we can detect a lot of different proteins which originate from different cells from the brain. For example, if you see on this picture, for example, there is a neuron and astrocytes and microglia and oligodendrocytes displayed on this picture. And normally there are some cells which are rich of these proteins, for example, neuron-specific annulase in the neurons, or S100B protein in astrocytes, or YKL40, GFAP. They are released into the cerebrospinal fluid by the astrocytes. Then there are other proteins like, for example, tau and NFL. They are always associated with an axonal damage, and myelin basic protein, MBP, is, for example, released by the oligodendrocytes. So this is something which is quite interesting, and the abnormal concentrations of these proteins, they can be measured in the cerebrospinal fluid. And now, because the techniques are progressing and there are very sensitive and reliable methods to measure these proteins in blood, so there is also something with blood on the horizon. And this is one class of the proteins and of the biomarkers which are also used for the to analyze cerebrospinal fluid in patients with prion diseases. So one of the first proteins which was extensively analyzed in the cerebrospinal fluid was the protein 1433. It was detected already 40 years ago, and at that time this protein was called P130, 131. It is displayed on this two-dimensional gel electrophoresis, and at that time there were no sophisticated possibilities to analyze these two-dimensional gels. So it was basically a visual inspection. But even on this visual inspection of the gels, it was very apparent that these proteins 130, 131 were seen in patients with prion diseases, but not in any other diseases which were analyzed at that time. So later on, it was clear that these proteins belong to the 1433 family. And then 1453 could be detected via Western blotting or later also via an ELISA. And today we know that these proteins are elevated in almost all patients with sporadic Creutzfeldt-Jakob disease, and to a certain degree also in genetic and variant and iatrogenic cases. Of course, when we look for the specific disease subtypes, we see that these proteins are elevated in MM1 disease subtype and in VV2. So there are two most common disease forms where we see the high levels of 1453 in the cerebrospinal fluid. And then there are other disease forms, disease subtypes, which are more rare, and they might be negative in the cerebrospinal fluid for these proteins. So if someone has a suspicion of Creutzfeldt-Jakob disease or prion disease, and there is a negative cerebrospinal fluid test, but the clinical syndrome is suggestive for prion disease, we should always consider, might this be a case of one of these rare genotypes? So this is just for the clinicians. There was a quite extensive debate on the 1433 specificity. So because 1433 is a neuronal protein which is released into the cerebrospinal fluid because the neurons are damaged, and this is probably not very much related to the prion protein and the pathogenesis of the prion disease, but rather a neuronal damage. So therefore the specificity of this test was questioned. And we did it in a multi-center European trial. So we collected samples from individuals, from patients, more than 6,000 samples, which were analyzed for 1433 protein. And then we analyzed the diagnosis in these cases. And then we came up with these data. So the specificity, when we compare prion disease, and we want to distinguish prion disease from Alzheimer's disease, dementia with Lewy bodies, or other neurodegenerative diseases, then the specificity is very high, 97%. So it is almost always quite discriminatory. But if we have to consider other neurological diseases like inflammatory diseases, paraneoplastic, stroke, epileptic fits, then you see in the middle of these bars, you see that the specificity is lower. It is not 97% anymore, it is only 82%. But still, it is for a clinical biomarker, it's still good. And what we always tell the clinicians who consult us for the potential diagnosis of a prion disease is, first we need to exclude this patient had an epileptic fit, has stroke, has an inflammatory disorders. And if these diseases are excluded, then the 1433 specificity is quite good, and it's also a sensitive biomarker. There was also quite extensive debate of another proteins which are elevated in the cerebrospinal fluid, for example, protein tau. And there are laboratories which suggest that instead of using 1433 test, protein tau should be measured in a cerebrospinal fluid, because it also has a quite good diagnostic potential. And you can see it from this slide here. In sporadic CJD, we have very high concentrations of tau protein, and it's quite good discrimination with Alzheimer's disease. And we do not see this with the phosphorylated forms of protein tau. So, for example, tau and phosphorylated tau forms are increased in Alzheimer's disease, but in sporadic CJD, we see increased tau levels, but no increase in phosphorylated tau forms. So there is a misbalance between in the elevation of both tau forms. So this was, and therefore this was suggested that tau protein, or the ratio between tau and p-tau, might be also good diagnostic parameter for inner cerebrospinal fluid, and even instead of 1433 test. So I do not completely agree with this view, and I would like to explain why. It is because I think 1433 test is the only one which we need to take for in suspected prion disease patients. So there is no other reason to measure 1433 other than someone is thinking this patient might suffer from Creutzfeldt-Jakob disease. So this is one reason. The second reason is tau protein, cerebrospinal fluid tau protein, is increased in many other conditions as well. And it is broadly used for the clinical diagnosis of Alzheimer's disease, for example. And what I would like to show you here is an analysis we have done in our center. It was in the neurochemistry laboratory at our center. It was not in the prion lab, but the other laboratory. And there we also get samples to measure for CSF tau. And when we applied cerebrospinal fluid, when we applied the cutoff for which we use for prion diseases to these data, you can see this in the chart that in all patients who had tau levels higher than 1,200 picogram per ml, only 25% of those samples were patients with prion diseases, but 23% of samples were patients with Alzheimer's disease or other acute and chronic vascular events, seizures, inflammatory, and so on. So by measuring CSF tau, also as a screening parameter, it might cause a lot of problems because, of course, all these other diseases like Alzheimer's disease and chronic vascular diseases and everything what is displayed here on this, they are much more frequent. And when we start to screen all these samples and suspect prion disease only because CSF tau is higher than 1,200 picogram per ml, we will have a lot of work to do. So therefore, I think I would not recommend to switch to the screening method or to the screening of cerebrospinal fluid levels for tau as a potential suspected diagnosis of prion disease. So I think it should be the clinical decision and the clinical definition of potential differential diagnosis of a prion disease. Yeah, so this is for the prion diseases, for the biomarkers which are commonly used on the in this field. And there's another class of biomarkers which is based mainly on the protein characteristics. So you know this concept of native and misfolded proteins, which then aggregate and use fibrils. And this is exactly what this other class of tests is using to detect the abnormal protein forms. And this is a development which was started with Claudio Soto, who described a method which he called protein misfolding cycling amplification. Yeah, I think almost 20 years ago. And this was I would say a revolutionary method because he really developed it and could demonstrate that you can amplify abnormal seeds into a detectable level. This was a quite important start of all these studies. And later on, this method was developed further and changed. In principle, it was changed a little bit, and by Byron Corgi, and he said that he called it a real-time quaking-induced conversion, and this is a methodology which is used today in many laboratories and. As I understand now, will also be available in Mexico soon. And the method works like this amplification model for nucleic acids, but it is for proteins. And when you look on the figure on the left side, then you see if you have a sample where we think there are prion protein seeds in it. So we take this sample, then we add recombinant protein, and we allow them to aggregate, to convert first from the recombinant, from the physiological form into a pathological form, and then it forms aggregates, and these aggregates are disrupted by high frequency shaking. After they are fragmented, so we get a lot of more of new seeds in this method, then the reaction continues. And once we have enough of these aggregated fibrils, we detect fluorescence because there is also a dye added to this sample, and if there are aggregates, then there is light which is emitted by the dye. So at the end we get this kind of curves, what you can see in the middle of the figure or also on the right side. And if there is an increase in fluorescence, we know that in this initial sample there was aggregated prion protein. So this is, I think, a quite elegant method to detect this abnormal protein, and it is also very, very sensitive because we can detect really amount of femtomole levels of the seeds. So it works quite well with the cerebrospinal fluid in patients with sporadic CJD, almost all patients with genetic CJD. It does not work very well with fetal familial insomnia and Gerstmann-Streusel-Scheinker syndrome, but there are also now methods to improve this. But in most frequent cases, this method allows us the detection of the abnormal protein, and it is very sensitive. So when you look on this figure, these are publications related to this field on the RT-QuIC with a little bit different changes in the methodology, but at least still RT-QuIC, and studies which have been performed or published until 2020. So today we have even more data, but at that time this was available, and you can see the sensitivity ranges between 82% to almost 96%. So this is a quite sensitive test, and also the specificity is very high. So we almost do not have any false positive results with this test. This is very good. So when we introduce this into the clinical practice, of course, we need to know how valid is this test? Is it affected by something? Are there any conditions we need to follow? And for this, we tested the RT-QuIC assay at different room temperature. So we kept the sample for up to 8 days at room temperature or at +4 degrees, or for up to 9 years at -80 in the fridge, and it was still very consistent results. So we had a high number of positive replicates. So apparently the temperature does not affect the validity of the test, and it is also not important to keep the sample on ice or on dry ice. So it apparently stays quite it's quite robust at room temperature. What is not very good is if the sample is contaminated by blood. So you can see it on the second graph. When we incubate the cerebrospinal fluid with blood, for example, with erythrocytes, or if we sonicate, if we pipette blood into the cerebrospinal fluid and sonicate, then the test becomes negative. And this is an important thing to know, so that if there is an artificial lumbar puncture, because sometimes difficult to do this, and there is the cerebrospinal fluid is contaminated by blood, then it is important to centrifuge it immediately to get rid of the blood contamination, because otherwise the test will turn negative and will be false negative. Yeah, so this is important for the for the clinical workup, for the clinic, for the clinic. And this is something just to mention that after we know that this RT-Quick is such an important test and such a good test, there was a consensus group working on it and on the diagnostic criteria, and also Michael was also part of this group, so we published it, and Brian Appleby also as well, and we published this criteria. So today we do not need to have this very complicated setting of the criteria, but a progressive neuropsychiatric syndrome and a positive RT-Quick will allow the diagnosis of a probable Creutzfeldt-Jakob disease or probable prion disease. So I think this is a big advantage we have now compared to what we had maybe 20 years ago. Okay, so, but there's also something else what we need to talk about this abnormal prion protein and the detection of the abnormal prion protein in human tissues, because now when we have all these fancy and sensitive techniques, we see that the abnormal prion protein is also detectable not only in the brain and in the spinal cord. It is also there in the various glands. It is detectable in lung. It is detectable in liver. It is detectable in skin. So it is apparently detectable in different tissues and fluids. And we were thinking at that time when we started these experiments, what I would like to show you in a second, we were just thinking, oh, let's try it. Maybe this is something what we can do, but we did not really believe that it will work. And the idea originated in a collaboration with Paul Lingo, who was a neurologist in our lab, in our hospital. And he was working with tear fluids for Parkinson's disease. He wanted to identify some biomarker in tear fluids in Parkinson's disease, and there are not so many people working on tear fluids at all, because tear fluid was not discovered yet as a potential source of, or potential platform for biomarkers. So we just said, oh, let's try and let's see what we can do. And then the work was done by Peter Hermann, Matthias Schmitz, and Susanna Correa. And what we have done, we just collected tear fluids, which is normally done with a filter paper. And it is, of course, you can imagine these are only microliters of tear fluid which can be collected. But even there, when we collect this tear fluid and apply it to the RT-Quick, we can detect PRP seeding activity. And this is quite interesting, because when we compare this with Creutzfeldt-Jakob disease cerebrospinal fluid, of course, you see the response in our reaction is quite fast. So it is here in the green. This is the green curve. So the signal goes up after 20 hours. And of course, this is not the case in the tear fluid. The increase is much slower. It takes much longer time to see a signal, but it is there. And this is something quite interesting, because we see this signal in around 80% of patients with prion diseases. So I do not believe that tear fluid one day will be the most important diagnostic test for the clinical diagnosis of prion disease, because for this, the cerebrospinal fluid is still more reliable, and it is still it gives us still more information than tear fluid can do, because of course we are excluding inflammatory disorders and all these type of things. But the tear fluids can be a source of biomarker platform for longitudinal studies. For example, when we are talking about potential therapies and we want to have a readout or in other settings, for example, when we are talking about early and presymptomatic disease stages. So therefore, I think this is something which is very interesting to follow, because tear fluid is also very easy to collect. It's non-invasive. It can be done multiple times. It can be done every day, and also even by a non-medical personnel. So this is something which is quite interesting. Another set of biomarkers which are now on the horizon are blood-based biomarkers, and there have been some publications on these proteins which were detected or are detectable in blood. And I just try to summarize this. There are some publications on protein tau and various tau forms, total tau, phosphorylated tau forms. Then there's a protein NFL, GFAP, S100B, beta-synuclein, YKL40, and the prion protein. And these proteins have been studied, some of them only in 1 or 2 publications, some of them a little bit more, like NFL or tau. But when we try to understand what these studies mean, then I try to put it here in this table just to give an overview what they are good for. If we look for the diagnostic accuracy of these proteins against healthy controls, neurodegenerative dementia, and CJD mimics, which means diagnoses which were presented like Creutzfeldt-Jakob disease, but CJD was ruled out, then it seems that, for example, tau proteins and NFL and, for example, beta-synuclein are performing quite good in the discrimination, in the diagnostic accuracy. Also, I must admit, we have more information on the protein tau and NFL because it has been published by several groups already, and we have only limited information on beta-synuclein because we have only 2 or 3 publications on it. So this is something which needs to be explored a little bit more, but it looks like that there are some biomarkers in the blood. When we want to know from these biomarkers, are they dynamic? So do they change with disease progression? Then there are still 3 biomarkers which occur like tau and tau forms, NFL and GFAP, and they change with disease progression. For example, they increase when disease progresses and they're dynamic, as I said. So which means that they might also be a potential biomarker once we want to monitor the disease, for example, during some therapeutic studies. So these could be biomarkers of choice in these diseases. But of course, we also want to know, can these biomarkers in blood tell us something about the prognosis of the disease? So, for example, are these fast progressing forms? Are these slow progressing forms? And it seems that tau and tau forms are quite good for that. And NFL and GFAP to a certain extent, but not very clear that they can tell us something about the disease prognosis. But for tau, we have some data that high levels are very clearly associated with a poor prognosis and with a short disease duration. And last but not least, preclinical. And this is what we all want to know, which biomarker is a preclinical biomarker, and so which means, for example, in genetic cases, can indicate that there is a disease or the pathology is already starting. And why do we want to know this? We want to know this because once we have therapies on the horizon and we know that there might be a pathology ongoing, we would like to treat these patients with these drugs, and this is of course very important, for example, for families with genetic prion diseases. Yeah, and it seems that there's NFL, it's performing better than other biomarkers, but it's still not very satisfactory. So we are still in search of blood-based biomarkers of neurodegeneration, prion disease, and I think this is something what is now on the agenda and what we need to do. And for this, I would like to present just 2 or 3 activities which are ongoing in our consortia, which address all these questions. For example, one is the International CJD Surveillance Network. Many of the members of today's presentations are members of this International CJD Surveillance Network. And for the moment, we just tried to revive the earlier activities which have been done in the field and which somehow stopped in recent years. So yeah, so we started again bringing people together, and we had already several meetings. Online, of course, for the moment, because there was no funding for that, but we will find a way to continue with it. And so we had general meetings, and we have also some focus meetings on neuropathology, on biomarkers, and also on biosafety. So, and everyone is welcome who wants to contribute to these activities, and we are very open, and we are a very open network for this. Another network which was created is the network which is called Prionet Risk, and this is a network which brings together all these prospective cohorts which are established in the countries. So one day I realized that there are many people doing preclinical cohorts, so they are following patients, or that here there are patient families who are how to say, organize themselves. So a lot of activities around, but they were disconnected. And then I was thinking we should bring people together, because of course the collaborative initiative and collaborative research is always much faster and much more productive. So we had the first meeting at the Prion conference, Prion 2020, which was in Göttingen. And since then we meet regularly and yearly, and either at the Prion conferences or also in other places. And we are working on different aspects. Of course, there are some issues with the biomarkers, also predictive biomarkers on the cohorts, on specific questions on genetic counseling, and all these type of things. So this is something we are doing, and yeah, everyone is welcome to join us. And just to see, I talked about the NFL as a potential biomarker, as a potential preclinical biomarker. This is a paper which was published from London, from Tim Mock and Simon Mead, and they analyzed several biomarkers in blood in patients with, or not in patients, in individuals with who were mutation carriers, and donated samples. And from these data, it is very clear that, for example, NFL is one of the biomarkers which increases very close to the clinical onset. And this is something what we could confirm in a mouse model. We had a collaboration with Olivier Andrioletti, an experiment with this infectious mouse model, and samples were taken during the incubation period and during the symptomatic stage, and you can see NFL increases very close to the clinical onset. But yeah, so this is, I think, the first step, but of course we would like to know more about the preclinical stage, so which biomarkers might contribute to the earlier diagnosis, which biomarkers might indicate disease onset, not some days just before the first symptoms start, but maybe give us more time to be prepared to start a treatment. So there's more things what we can contribute in this. And I think one of the advantages to study, to contribute to Prionet Risk is also exactly what we can do there. We can collect samples, we can follow patients or individuals and patients, and we can then identify biomarkers which might be more suitable than NFL, probably, hopefully. Yeah, and just because time is running out, I just would like to present you this activity, because I've heard that there will be Arctic laboratory in Mexico. So we also established a program which we called Prion Pro, and this is a program which is dedicated to increase the or to improve the sensitivity and specificity of the biomarker tests, and also to help people to establish the laboratories for Arctic testing. And for this, we sent samples to participating laboratories, and we do ring trials just to compare how valid are the tests, because of course everyone is doing it in a different way, and then it is very important to have a comparison also for everyone. And in 2023-24 ring trial, we had 13 labs participating from 11 countries, and now 25, we have already 18 labs from 14 countries. So if there is a need and anything to do, please join us if you want to participate in this trial activities. Yeah, and that's it. I have not done these things. These are many people who contributed, and these are people working in different fields, from physicians to neuropsychologists and laboratory people, biomarker development. So everyone is involved in a way or the other. And thank you very much for listening. I don't know if the questions are allowed, but I will be happy to answer them. Thank you.
Thank you, Inga, for this great presentation. We have one, I will say, interesting question from the audience. There is a dentist who is asking about some prion findings in the mouth, or if there is a way of having some diagnostics test through the mouth. I don't know if you heard.
Yeah, so this is there are some publications that it can be that the disease can be transmitted via dental route. That's true, and there are also earlier publications in animal models which confirm this. But as far as I know, it did not happen in humans. So we do not have now any clinical or other evidence that it can be transmitted via contact with dental procedures. So in terms of a test, there is, as I told you now, there are some possibilities to detect abnormal prion protein in various biofluids and organs. And there are also groups working on saliva, for example, could also be a source of biomarker for this. But saliva is a little bit more complicated than tear fluids, for example, because there are much more proteins there. Tear fluid is a very pure tear fluid. There are no cells and nothing there, so it can be tested very easily. But I think it will be saliva will also come one day as a test.
Okay, thanks. And we have one last question from a geneticist who's asking something about the, I don't know, maybe it's Arctic Week, because he's asking whether you could calculate like the speed of aggregation of the proteins or the speed of transformation of the proteins.
Yeah, this would be ideally if you could do this. For the moment, this that several people tried, and we also tried to do some estimates from the curve. Is the curve, the curve and the speed of aggregation, is it somehow correlated with the fast progressing forms or with a slow disease progression? So ideally, you would think if it aggregates fast, it would be a prognostic factor. But we could not really find this. And I think it is, for the moment, at least not very clear, because these are only, we are detecting 3-4 molecules, and I don't think this is something we really can have this type of a readout for the moment.
Okay. Thanks, Inga, for your presentation. I don't know if you already asked me about whether to join in some of the meetings. If I will again thank you for the invitation, and please include Mexico in the following Priones. So we'll be happy to with the few data that we may have.
Victor, I already put you in our list. Yes, and you have seen you are already on the map.
Thank you so much. Thank you so much. So we will have to move on for the agenda. We already have Dr. Eric with us. So Eric, if you're ready for presenting.
Yeah, absolutely.
Thank you so much, Eric, for being here with us. Let me just do a somewhat quick introduction of you in Spanish for the uh, audience, and then I will give you the microphone. Ok, so el día de hoy tenemos también en nuestro simposio como parte de las estrategias para encontrar algún nuevo tratamiento para las enfermedades por priones al doctor Eric Minikel. El doctor Eric Minikel se encuentra investigando en el Broad Institute, que es un centro de colaboración entre universidades, entre ellas Harvard y el MIT. El doctor Eric inicialmente era un investigador en otra área en Harvard y posteriormente migra al estudio de enfermedades por priones, donde intenta o está trabajando de manera muy intensa en buscar nuevas estrategias terapéuticas para las enfermedades por priones. Ha tenido también un enfoque muy importante en el estudio de las variantes genéticas de las enfermedades por priones, donde tiene un blog y ya ha publicado muchos análisis en este sentido y que también se han reflejado en publicaciones científicas a nivel mundial. No quisiera entretenerlos más, me gustaría dejarle la voz de una vez al doctor Eric, donde nos va a hablar sobre estrategias para buscar nuevos tratamientos para estas enfermedades. Thank you, Eric, again for being here with us. So we're ready to hear you about the new therapeutic approaches for prion diseases.
Awesome. Thank you so much for having me. Really glad to be here. Here we go. How's this? Good.
We can see your presentation and we can hear you very well, Eric.
Lovely. So I have a half hour, right? I'm going to give kind of a overview of some of what I presented at the Prion conference in Brazil earlier this month. You know, for those, I spoke at this symposium last year, but for those who don't remember, so this is my wife, Sonia. She has a genetic prion disease mutation, D178N. We lost her mother to genetic prion disease 15 years ago, and we changed careers to become scientists to develop a drug for her before it's too late. So this is our lab. We have 17 people now here at the Broad Institute, and our goal is to develop both a treatment for symptomatic patients and a preventive for patients at risk. And I want to spend a lot of my talk today arguing why we should focus so much on PRP lowering as the strategy for prion disease. Some people will refer to prion disease by these historical names like CJD, FFI, GSS, but at the molecular level, it's all one thing. There's one pathway of disease. You need to have the normal prion protein in order for it to misfold. And if we attack the normal prion protein, that unites all these subtypes of prion disease as one thing. And so then it no longer matters, are you sporadic or genetic or acquired? It doesn't matter what subtype. It doesn't matter how it presents. And to a large extent, it's effective even different disease stages, right? Treating early or treating late. It's all the same therapeutic hypothesis. So we're already a pretty rare disease, but going after the root genetic cause of the disease gives us a greater likelihood of success and unites us as at least less rare than we could be if we considered these separately. And people always ask, isn't there a reason we have normal prion protein? Does it have some job? And yeah, there is a neuropathy in aged animals with no PRP, but it's really quite mild. And in the face of how severe prion disease is and how clearly the scientific evidence tells us we should lower this protein, this should not stop us from developing PRP lowering drugs. So the reason we focus on PRP as target is that any which way we study this disease, we get the same answer. PRP is the cause of it, right? So you talk to a neuropathologist who looks at the brains of prion disease patients, they'll say that what you see in those brains is deposits of abnormal PRP. You take a biochemical approach and purify out the infectivity from brain material. That infectivity is made up of PRP. Genetic variants that cause prion disease, like my wife's mutation, are in PRP. And then the risk factors that control risk of sporadic prion disease are also in the PRP gene. And we have really good animal models in prion disease. This is a disease that naturally affects many different mammals, right? Camels and goats and sheep and deer and elk and cattle and mink all have naturally occurring prion disease. And so prion disease is really well modeled in mammals, including in mice. And when we study this in mice, the answer we get is that PRP lowering works really well. And at this point, this is just a summary, right? Each row of this table could be its own presentation. but this is just a summary of all the studies that we have presented or published. So we've done 4 different species, 7 different PRP amino acid sequences, 6 therapeutic drug modalities, plus genetic perturbations, and 12 prion strains. And in every model that we test, PRP lowering is helpful. And by doing all those studies, we've learned under what circumstances does PRP lowering work best. So one thing we've asked is, does it work against all prion strains? And it seems that it does. How well it works if you lower only 50% varies. So it's really good against some strains and it's pretty good against other strains, but it's always dose responsive. So if we can lower deeper than 50% and get close to 0, we can address all of these strains. We've looked at disease stage, and it works pre-symptomatically. It delays onset of disease in animals. After symptom onset, it slows progression. One thing we found is that it's very difficult to do any benefit for animals that have less than 3 weeks left to live. And that's probably just because PRP lowering needs time. So I'll talk about that in a minute. And we've also found that if you get 50% lowering of PRP, it's kind of the same whether or not that's 50% in every cell, which is about what we do with an ASO, or 100% in 50% of cells, which is about what we do with a zinc finger repressor, another drug modality we tested. So that means there's a variety of different approaches we can leverage, and maybe someday we'll even combine them, but we obviously have to show they work alone first. Another reason to focus on PRP lowering is we can measure that we did it. So in common diseases like Alzheimer's, where there's tons of money, people will do huge trials of hundreds of patients for years and years, spend hundreds of millions of dollars without having any idea whether the drug does anything. But in a rare disease like prion, no one's going to make that kind of investment without knowing upfront that the drug is active. And we need to prioritize putting our best bets on things that we know are having an effect. And so when we lower PRP, we're lowering it in the brain, and we've shown here in rat and here in monkey that that results in it being lowered in cerebrospinal fluid, or CSF. So therefore, we can measure that we did it. And in pre-symptomatic people at risk, we've measured their CSF PRP over many years, and it's very steady. Over 6 years, people are at exactly the same CSF PRP level they started. So if we lower that by half with the drug, we should be able to see that difference really clearly. And we also know that there's a bit of a lag time. So when we lower the PRP RNA, it goes down fast, but the protein takes a little extra time because of its half-life. So that probably explains why we need 3 weeks to actually benefit an animal. And these are just some data to show that. So when we treated animals very late in disease, when they had 9 days left, we could not make an impact. But when we treated with 25 days left, we were right on the edge where we could help some of the animals, but not all of them. And if we treat with 40 days left, we can usually benefit all of the animals. So those give us some learnings that we can take into how to apply these drugs. And there's basically, I think, of 3 categories of approach or drug modality that we can use to lower PRP. So we can target the DNA. We can turn off production of PRP starting from the DNA, from the gene. To do that, we need a gene therapy that is going to be a large protein complex that binds or edits DNA. That large protein complex is not going to just get into the cells. The way that you deliver those kind of things into the cells is using a viral vector. So you encode the DNA for that complex into a virus and you inject the virus. There are now engineered viral capsids that, in mice at least, can cross the blood-brain barrier. And so we think a one-time dose into the blood could give you lifelong editing of the gene and a permanent therapeutic effect. Whether those vectors work and are safe in humans, we still don't know. This is really recent technology, but it's a reasonable shot on goal. It's a type of drug we can try to develop. And what you put in those vectors, we've so far tested 3 options. One is to edit the sequence of the DNA with base editing. One is to do epigenetic marks, so methylating the DNA to turn off transcription of DNA to RNA permanently with CHARM. And another is a zinc finger repressor protein complex that binds the DNA, and as long as it's there, it prevents the RNA from being transcribed. So these are all things we've tested in mice and they work. They're all still some years away from potential clinical application. Slightly more immediate, we can use oligonucleotides. So these are short sequences, maybe 20 base pairs of chemically modified DNA or RNA. Historically, these have not been engineered to cross the blood-brain barrier, so they're administered into spinal fluid, so through a lumbar puncture or intrathecal injection. And they last a pretty long time, maybe 1 month, maybe 4 months. Eventually, they need to be redosed. The ASO developed by Ionis Pharmaceuticals now has a fully enrolled clinical trial. We're hoping in the next few months they will announce some results from that trial. Our lab also developed a divalent siRNA, which I'll talk about at the end of this presentation. We hope that we'll be dosing the first patients in the US, again, within the next few months. Finally, there's one really exciting effort. There's a biotech company called Gate Bio that is developing a small molecule that they think could be a pill that you would take every day. And that small molecule binds the protein just as it's beginning to be produced and prevents it from being translocated through a pore onto the cell surface. And therefore the protein gets degraded. So it's at the moment of translating from RNA to protein that it blocks. So I think we need all of these approaches and more. I would love to have 10 more approaches in the race to lower PRP. And I think we have already done as many experiments as we need to do or should do in animals to de-risk this, right? I think it's really clear that prion disease depends on prion protein. And if we lower prion protein, we're going to slow it. That biology is really fundamental to everything we understand about this disease. It makes sense with the genetics, the biochemistry, the neuropathology. In a variety of animal models, it always works. And these animal models are authentic. It's the same disease that humans get. So I think the probability that we're wrong about the biology is extremely small. I don't think the biology will be the reason we will fail. But there's still lots of ways to fail. And I think the bigger risks now are drug-specific factors. So every drug has toxicology, and every drug, for the most part, is going to have some limitations in biodistribution. So where does it go? Does it get to high enough concentrations in the body? So any one of these drugs could fail for those two reasons. And then there's another reason, which is true across all of these different drugs, which is how we design and execute the clinical trials. So when we design a clinical trial, you want to show that the drug benefits patients, and that becomes a statistical question. And so do we have enough patients? Were they progressing fast enough that we can show a difference with the drug, but not so fast that the drug was too little too late? Did we have the right mix of types of patients? Were they balanced between the groups? There's so many trial design questions that go into making sure that we have the ability to detect the drug's effect in a trial. So we believe that the solution to the first one is to have more than one drug in development. And the solution to the second problem is we need natural history data describing how the disease normally progresses, so that we can use that to model out our clinical trials. So I'll show just a little bit of very, very preliminary data. I think I saw Brian Appleby on the call. He's been a heroic and amazing collaborator. He really shares, and he shared with us a lot of data from the US Prion Surveillance Center that have been really helpful. And what I'm showing here is the median. So these box plots are median in the middle, and then interquartile range, so 25th, 75th percentile, of the time from when a spinal fluid sample got to the surveillance center for diagnostic testing until the patient died. And you can see that for MM1, which is the most common subtype, it's about a third of patients. On the median, it was only 20 days from the CSF testing until the patient passed away. And then there are a number of more slowly progressing subtypes, tremendous amount of variability both within and between subtypes. So that's a challenge. The other thing you'll notice is that when we say MM1, the 1 is a PRP scrapey type. It's a type that is only determined on autopsy. So when the patient is still alive and enrolling in a clinical trial, we don't know if they're are they MM1 or are they MM2? And you can see here that there's a 10-fold difference in survival. 192 days for MM2, 20 days for MM1. Now, some of the difference can be predicted by CSF total tau, which is a marker of neuronal degeneration, neurons dying. And you can see that total tau is some indicator of how rapid the disease is progressing, and it's been shown to predict how much time patients have left. It is much higher in MM1 than in MM2. So there are other factors besides subtype that can tell us something about the patient's progression. And in fact, if we wanted to use CSF total tau to tell which patients are MM1 and which are MM2, it's not perfect, but it's a pretty good discriminator. Other people have used MRI to classify the subtypes in living patients. This is almost as good. The tau is almost as good as MRI. And a lot of variables predict survival time in prion disease. All of the different subtypes have their own characteristic. Progression rate, sex, age, and total tau are all also significant. But everything I just showed you was time from CSF until death. But in fact, many patients, even before they die, have no quality of life left. And what we want is to not just show that a drug keeps people alive, but that it gives them some quality of life, keeps them alive in a state that their family is glad to have them around, right? Sad to say, but this is what we're up against in this rapid disease. There were plenty of moments in my mother-in-law's illness when I'm not sure it would have benefited her or the family to keep her alive longer. And so we need to think about designing trials where we enroll people early enough that the drug has time to benefit them and that benefit means something. And then we need to follow them and show that we kept them in a state where they were able to function for some period of time. And the clinical rating scale, MRC scale, that was developed in London and that Brian has used in his TAP-CJD study for years now, provides a potential way to do this. And so what I'm showing on this plot is each of these lines represents one patient's journey. So from their initial assessment, how did their MRC scale decline over time as they came back monthly or bi-monthly for assessments over Zoom with Brian and his team? And one way to slice this is I told you from the animal studies and from the half-life of the protein, we think that we really need 30 days to get time to effect for a PRP lowering drug. And I showed you in this plot that there's plenty of patients who do not have 30 days left, right? In fact, for the most common subtype, the median patient only has 20 days left. So in order to benefit patients, we need to do the clinical trial in patients who have 30 days left. If you draw a line at MRC of 15, there are a few exceptions, but for the most part, patients with a 15 or greater have got 30 days left to live. And then you could ask, well, how long should we follow them? Both because we want to set a threshold where people still have quality of life, and because how long patients actually survive depends somewhat on decisions their family makes. Some families will keep a patient alive on life support. That doesn't really tell us anything about the biology of their disease being different than other patients, but on paper, their survival time could be months and months longer. And that variability, one patient went on life support and one didn't, could be really bad for the statistics of a clinical trial. If it looks like there's a lot of variability in both groups, the drug treated and untreated people are overlapping, they kind of look the same. What you want is to get rid of all that variability and just look at the biology of how fast did the disease progress. And I submit to you that most of the time when we see a patient sort of MRC score flattens out, they reach a low level and then they stay at that low level for a long time, but alive, that usually happens below a 5. And I don't know that those patients were necessarily on life support, but it does seem like there is this phenomenon where some patients will progress rapidly through the middle of the scale, and then they kind of hit a very low level where I think they probably don't have much quality of life left, and they'll be alive for a lot of additional time. So it's really from 15 to 5, I think, that you see a pretty fast linear decline in people's function. And maybe that's where clinical trials need to run, is people start with at least a 15 and you follow them until they've hit a 5. And that allows you to compare, did people progress more slowly on drug than off of drug? So again, these are just really preliminary analyses. We just got this beautiful data set from Brian, and we're working on analyzing it now. But the vision is that our lab wants to put together a collaborative publication with Brian and all the other folks worldwide who are able to share these natural history data with us, and sort of make a guide to how we can do clinical trials in prion disease that are meaningful and well-powered. And I think that at this point is the bigger contribution to the potential for success in developing a prion disease drug than just finding another molecule. We've already got a number of molecules in development. Now what we need is the right way to test those molecules in the clinic. So I will give one quick update before I sign off on a drug that our lab developed. So this is the drug. It's a intrathecal, that means injected into spinal fluid at the base of the spine, oligonucleotide drug. It's designed to lower PRP. It was developed in our lab in collaboration with University of Massachusetts as a US federally funded grant project. And we found it worked really well in our mice, and we wanted to advance it to humans. As of last year, we manufactured these drugs. You can see it's actually labeled for human investigational use. It was really thrilling to see these vials, and they're now sitting in the freezer waiting to be dosed. We got clearance from FDA that we are allowed to proceed with the human trial. But of course, running a clinical trial is complicated, and having permission is not the only rate-limiting step. We also needed money. NIH awarded us a grant in August that will allow us to run that. After getting that grant, we were able to start all of the activities, very complex activities, setting up a database, getting ethical approval, training all the sites on the procedures that will allow us to run a clinical trial. So at this point, the ethical protocol is submitted. It's in review at the hospital. We have an investigator meeting next month where we will train everyone. We hope that early in the new year, we might activate a site and begin to dose patients. Unfortunately, we're pretty limited at this point. So it's only US sites because this trial is funded by the federal government. We don't have the ability to have a site in Mexico. I'm sorry, I would love to. I'd really love to work with Victor on this. I really want people like Sonia to be on this drug, but FDA does not want to let us treat pre-symptomatic patients yet. Right now, only symptomatic. We could only afford to do a single toxicology dose in our animals. So right now, we're only allowed to give humans a single dose. We can't repeat dose them. We can't do any compassionate use programs. There's a lot of things we can't do. But our goal is to just get the very first human data. By the way, for anybody else working on drug development, we made our regulatory filings public. There's very few examples of investigational new drug applications public. So we hope that ours can be a resource to people. Just to show a little bit of the animal data, this is a dose in mice, 52 micrograms, that we think corresponds to the top dose we could reach in a clinical trial. You can see that even a single dose gives us 50% PRP lowering after 30 days. So on a time scale that is meaningful in prion disease, we're having a significant impact on the protein with just one dose. The big limitation, I would say, is that because it's administered into spinal fluid, it's not going to distribute perfectly evenly throughout the brain. And so in the toxicology studies in dog and rat, we saw great concentrations of drug in the cortex, the cerebellum, and in the spinal cord. But in deep brain, such as thalamus, caudate putamen, we didn't see as much drug as we would like to. There's some, and we may see some PRP lowering, but it's not going to be super deep PRP lowering. And I think this is a limitation also of Ionis's ASO, and really of any drug administered into spinal fluid. And people like to think that they can de-risk that through animal studies, and I disagree. I think you don't know anything until you've treated humans, because this is from the Museum of Science in Boston, about a mile from where I'm sitting here. This is an actual monkey brain. This is an actual human brain. People think monkeys are a, quotes, large animal model. There is no large animal model except humans when you're talking about the brain. This monkey brain is 5% the size of a human brain. And so if you can get into deep brain regions a little bit in monkey, that's great, but that doesn't mean you'll get any concentration in those brain regions in human. And I don't think that as of today, we have the autopsy data from previous clinical trials to prove whether drugs really reach those parts of the brain. So this is all a hypothesis that we need to test in the clinic. There's only so much we can do with animal studies. The good news about PRP is that PRP lowering is very dose responsive, and even a little bit of lowering is helpful. So if we lower PRP 50% in the cortex, and we lower it 10 or 20% in the deep brain, that's still better than not lowering it. That's my guess. I think it's worth testing, but we also need to do better. So for the clinical trial that we're running, it's a really small trial. We hope we can show that it lowers PRP and hopefully that it's safe. It also allows us to generate some data about what it looks like to run a clinical trial in prion disease. And because we're academics and we're patient scientists, we hope we can share those data publicly to help everyone. We're planning to enroll an observational arm of up to 15 patients, and then 15 patients will be treated with drug in a single ascending dose fashion. So each patient just gets one dose, and as we establish the safety, we will move to higher and higher doses. The main goal, again, is just to show that we lowered the protein. This is not enough patients to show that we slowed down the disease, but the idea is that this trial maybe could generate enough data to motivate a large future trial. Once we know the dose and once we know that the drug is safe enough. It's very interesting thinking about how to run these clinical trials, right? People who participate in these trials are not necessarily doing it to benefit. There's no human data to suggest a benefit. I think a lot of them are doing it just to help research. And there are a lot of risks. The toxicology studies for our drug looked very clean, meaning we didn't see toxicity. But nonetheless, if you receive this drug, you might be excluded from a future trial of a different drug. The drug could keep you alive in a state with no quality of life that might not be a benefit to you. And it's also a lot of trips to the hospital, a lot of interaction with the broken medical system at a time when you don't have a lot of time left. And that may not be how you want to spend your final weeks. So there's a lot of real challenges here. And I think that all the people who participate in clinical trials, despite all of this, are absolutely my heroes. And I'm so grateful to the patient community for being willing to work with us on these experimental drugs. And I think what we owe to them is to share data. And so I've been going around asking other academics, but also companies working on prion disease to share data. And I don't mean just publish a paper, give us a table, give us a plot. I mean to share individual longitudinal patient data. That's what's needed for modeling out clinical trials and figuring out how to develop drugs. And I believe that everybody who's working in drug development in prion disease, they owe that to the community to share those data with us. So that's my presentation for today, and I'm happy to take questions.
Thank you so much, Eric, for this presentation. And from the previous presentation, we've heard some of the questions that you already talked about. How to develop a clinical trial in a rapidly progressive dementia, which is also an ultra rare disease. How to have a sufficient amount of people enrolled in the clinical trial, how to follow them up. And I will also add this question. What will be the best way of determining that the drug is working in the clinical trial? Is it only because due to the PRP detection by RT-QuIC? Is there any other way of doing that? If they already enrolled and if they already have symptoms, is the MRC diagnostic criteria useful for this? Or
Yeah.
will there be any other way of establishing that this drug is really working? This or any other drug?
Yeah. Thanks. It's a great question. So how we show that a drug is working depends on the development stage of the drug. So in a phase 1, first in human clinical trial, what we need to show is that the drug is safe enough. It doesn't have to be perfectly safe, but it needs to be safe enough that we can administer it to patients and do clinical trials. And we need to know that it's lowering prion protein in the spinal fluid. And we do that, and it's not RT-QuIC. I don't think that we're going to turn a positive RT-QuIC negative, because the prion titer in the brain is absolutely enormous at end stage disease. And even if you lower PRP 50%, even if you lowered it to 0, I don't think that the RT-quick seeding is going to go away very quickly in the brain. So we're measuring just normal prion protein to show that we reduced the amount of the protein that's being produced in the brain. And that will tell us that the drug is doing its job. It's doing what we designed it to do, which is lower PRP. So from a phase 1, we just want to see safety and lowering PRP. In a pivotal trial, so a phase 2 or 3 that is designed to get approval of the drug, then we need to show a clinical benefit. So this is usually the standard with FDA, is drugs are approved based on clinical benefit. And clinical means it has to be something about how the patient feels, functions, or survives. And so there, it's not enough to show a molecular change. And I think the obvious thing to do in prion disease is the MRC scale. I think that's the best validated scale. Ionis has been using it. They've published a manual on how to administer it. Brian and others have shown that it has good validity and inter-rater reliability and other properties we would like. So my guess is that pivotal trials will be done using the MRC scale as an endpoint. It is a rare disease, but patients are very motivated, and I think we'll have no trouble finding enough patients. I think the bigger challenge is that those patients may be very heterogeneous. We will get some patients who have subtypes where they progress quickly and others that progress slowly. And that's why I think we need to be really thoughtful about how we select patients for the clinical trial and how we balance the different arms of the trial.
Thanks. I don't know if Francisco, ¿querías preguntar algo? Do you want to ask or say something, Francisco?
Solo agradecerle otra vez aquí a Eric, la verdad, su participación aquí con nosotros. Thank you very much, Eric, for being here with us. It's a pleasure here. Always you have your compromise with this foundation. Thank you very much.
Thank you for having me. Really grateful to be here. It's wonderful to talk to everyone.
Yeah, Eric, I have another question. I guess you already mentioned that this approach works with every type of prions. So is that what you said? That it's
Yeah. From
so,
first principles, all prions are made up of misfolded PRP, so PRP lowering should work against all of them. And in our animal models, we've tested 12 prion strains, and it always worked. So
okay.
as far as we can tell, it works against every type of prions.
Okay, so I'm thinking about the clinical trials that are used for Alzheimer's, for the genetic cases where they typically enroll the preclinical cases. Do you think these preclinical cases for genetic CJD will also be a very good population for trying to determine the response to the drug, or how good it is working?
Yeah. So pre-symptomatic people, meaning people like Sonia who have a genetic test report, know that they have the mutation, but no symptoms. I really want to do clinical trials in that population. It's our greatest opportunity for benefit because people have full quality of life we can preserve. CSF PRP is really stable in that population, so it's really easy to see if we lowered the protein, and they have no other health problems in general. So it's really easy to tell whether the drug is safe. Whereas I think in symptomatic patients, it's harder to tell what's disease progression and what is drug safety issues. So I think it's a great idea to do trials in pre-symptomatic. I really want to do it. Unfortunately for this drug, FDA has not yet given us permission. I hope they will. I think how you show that the drug is effective is harder in pre-symptomatic, right? Because if people aren't sick, you can't show the drug makes them better. You can't show that it makes them progress slower. They're not progressing because they're not sick. So I think that's where we will probably rely on alternative regulatory pathways, such as accelerated approval in the US to get approval based on a biomarker. And then in the long run, we will have to follow patients on drug over, a decade or more and show that, indeed it has kept them alive longer than would be expected.
Okay, thanks again. Eric, for your great presentation, also for the effort you're doing for the whole Prion community, to the families, obviously, and to the patients that have gone through this. I guess we'll have to move on with the agenda. Thanks again, Eric, and I
Thank you,
guess
Victor.
see you soon.
Honored to be here. Appreciate you having me. Take care.
Thank you so much. Yeah, so I guess we'll move on with the agenda, and let me see if Dr. Gabriela Valle is already here. I guess she's not connected.
Hola,
Ya estás conectada.
sí, doctor, solamente me falta compartir mi presentación, ¿le parece? Va a ser muy rapidito, no
Sí.
más de 15 minutos tal vez.
Ok, muy bien. Nada más que no te veo aquí en la conexión, no sé a través de qué red te enlazaste, pero adelante. Si puedes ir subiendo ya vi. Si puedes ir subiendo tu presentación, perfecto, ya lo tenemos ahí. Vamos a ver ahora un tema que también es muy interesante, muy importante, que es cómo atender al cuidador. Sí, el cuidador es un elemento muy importante en este tipo de
Sí,
enfermedades,
gracias.
tiene una gran sobrecarga. Y para este fin vamos a escuchar a la doctora Gabriela Valle, que tiene un doctorado en salud mental, una maestría en salud pública, doctora en salud mental, y precisamente cuyos trabajos de investigación han estado enfocados con los cuidadores para enfermedades neurodegenerativas o enfermedades con algún trastorno mental. Ella es investigadora en el Centro Universitario de los Altos, y nos ha estado apoyando también con este abordaje del cuidador para enfermedades neurodegenerativas. Sin más, le dejamos la voz y el micrófono a la doctora Gaby Valle. Le agradecemos su presencia y estamos listos
Gracias.
para escucharte, Gaby. Adelante.
Sí. Muchísimas gracias a todos. Gracias por estar presentes. Gracias, doctor, por su invitación. Bien, como ya lo menciona el doctor Víctor, soy doctora en salud mental, un profesor investigador del Centro Universitario de los Altos. Y básicamente hago este trabajo colaborativo, pero desde la perspectiva de las personas que cuidan a otras personas que tienen o padecen algún trastorno mental. Bien, parte de esto, como la sobrecarga es una de las manifestaciones que más presentan los cuidadores primarios informales. Hablamos de cuidadores primarios informales a quienes son familiares directos o indirectos, o incluso algunos vecinos que apoyan con el cuidado del paciente con algún tipo de trastorno mental, ¿no? Sobre todo en enfermedades crónicas, ejemplo, o otra enfermedad mental crónico-degenerativa como lo es el Alzheimer. Estas tareas suelen ser muy desafiantes, sobre todo en estas personas, en estos cuidadores que no tienen el conocimiento adecuado. En enfermería somos cuidadores formales porque ya tenemos una serie de técnicas y procesos que nos llevan al cuidado del paciente. Es más específico, más protocolario, tenemos una serie de pautas a seguir, protocolos, etcétera. Y cuando lo hace una persona que no tiene este tipo de educación, lo hace de una manera, digamos, algo lírico, algo que se aprende conforme va avanzando la enfermedad, ¿no? Esta sobrecarga que padecen los cuidadores primarios tiene efectos en la salud mental y por lo tanto desequilibra el bienestar de los cuidadores. Por eso es importante que nosotros identifiquemos cuáles son estos factores para poder contribuir y tener nuevas estrategias que sean más efectivas para proteger la salud mental y física de los cuidadores informales. Bien, como una definición hacia la sobrecarga del cuidador, nos habla que estos pacientes van a presentar un estrés muy significativo y sobre todo van a verse agotados. Ese agotamiento no solamente va a ser físico, sino también emocional. Y obviamente una cosa va a llevar a la otra. Recordemos que si no hay salud mental, tampoco va a haber salud física. ¿Sí? Entonces, esta es una parte importante, el por qué nos llama la atención hablar sobre esta sombra que está aunada hacia los pacientes que tienen un padecimiento de una enfermedad crónico-degenerativa, ¿no? Podemos ver al paciente enfermo, sin embargo, podemos dejar a un lado la salud mental de el cuidador primario, ¿no? Podemos verlo como una obligación, podemos verlo como que es su hijo y le toca, pero nos olvidamos que tiene ciertos aspectos, perdón, ciertos aspectos y ciertas características que no cumple de una manera profesional y que en ese esfuerzo extraordinario que está haciendo por el cuidado va a estar inmerso el desgaste como tal, ¿no? Y a esto nos lleva pues que tenemos causas comunes de esta sobrecarga, ¿no? El tiempo que se le tiene que dedicar, la ausencia del demás apoyo de las personas, las exigencias físicas, porque incluso hay pacientes que necesitan ser apoyados para que puedan moverse de un lado a otro, algunos que ya perdieron la capacidad de marcha y es necesario auxiliarlos a los movimientos, el impacto emocional también de estar viendo cómo su familiar se va degenerando continuamente, ¿no? Entonces, estos son los principales factores donde, o que contribuyen más bien a esta carga, a esta sobrecarga del cuidador, ¿no? ¿A qué nos lleva todo esto? Desde lo físico y lo emocional termina evocando algo que puede ser fisiopatológico, ¿no? En el cerebro obviamente va a haber un estrés crónico, va a haber depresión, va a haber ansiedad, agotamiento cognitivo, alteraciones del sueño, eh, todo lo que pueda estar inmerso dentro de que puede dejar de funcionar de una manera habitual o normal el cerebro del cuidador. Entonces, ¿por qué? Porque, bueno, sabemos que existen algunas conexiones donde aquí si falla algún lado, pues va a empezar a fallar todas las demás y esto va a ser como una bola de nieve, ¿no? Tengo olvidos, ya no tengo buenas relaciones interpersonales, me estreso porque tengo olvidos, me estreso porque tengo malas relaciones interpersonales y entonces va a ser un cúmulo de situaciones que van a seguir aumentando el estrés y por lo tanto también van a ser estos estímulos de cortisol que van a generar otros procesos inflamatorios y que después van a tener problemas musculares, problemas gástricos, cefaleas, etcétera, etcétera, ¿no? Estos aspectos de vínculo emocional que se generan por este cuidado excesivo hacia el paciente que padece algún trastorno, nos da, obviamente, no todo es negativo, algunas pautas positivas y otras tantas negativas. Si hablamos de lo positivo y que puede ser en un lapso más corto, lo que se tiene como algo positivo, porque es el inicio, porque mi paciente todavía no está tan degenerado mentalmente, porque puede ser millón de situaciones que todavía abonan o que lo hacen desde el amor y el compromiso. ¿Sí? Es papá, es mamá, es un hermano, es mi abuelo, es mi abuela. Entonces se hace desde el amor y el compromiso. Y eso nos lleva a algo positivo que puede ser la satisfacción personal de saber que tenemos este sentido de utilidad o de pertenencia, ¿no? Y obviamente que también se fortalece la relación. Pero como todo, o sea, todo esto en exceso puede llegar incluso a generarnos algunas otras cosas negativas como el estrés, la ansiedad, el sentimiento de culpa, aislarnos socialmente y una ambivalencia emocional entre amor y odio. ¿Por qué nos puede llevar a esto? Porque es posible que el cuidador, desde su trinchera, esté manifestando este compromiso que tiene, pero al no tener una red de apoyo que esté más fortalecida, él va a autoadjudicarse todas las actividades en torno al cuidado del paciente, lo que lo hace que sea este vínculo algo complejo y multifacético, y que nos lleva después a generar más sentimientos negativos, porque obviamente hay un desgaste. Si hay un desgaste emocional, la negatividad va a entrar. Y aunque lo sigan haciendo con mucho amor, con mucho compromiso, que les dé mucha satisfacción personal, va a terminar yéndose al otro extremo, porque ya no tiene esa capacidad o esa resistencia, ya rompió, y a donde va es a aislarse, a sentirse culpable porque ve que el paciente se degenera y cree que es su culpa, que no le ha dado bien cuidado o no lo ha cuidado muy bien, a tener estrés y ansiedad, en pensar en los easy, y si no le estoy dando bien el medicamento, y si lo que le di de comer le hizo daño, y eso nos empieza a generar ansiedad. Entonces, por eso viene como esta ambivalencia emocional. O hay algunos que lo han externando, sobre todo en algunas investigaciones donde se aplica el test de sobrecarga de Sarit, viene una pregunta muy particular, ¿no? Que a veces desearían que no estuviera vivo, digamos, su paciente, y la verdad es que lo contesta muy sinceramente: descansaría yo también. Entonces esa es la parte donde existe esta ambivalencia emocional como tal, ¿no? Bueno, ya casi terminamos. Por lo tanto, este desgaste, si lo vemos desde este perfil negativo, este desgaste nos va a llevar a que el paciente tenga un agotamiento extremo llamado burnout, que la sobrecarga nos lleve a la ansiedad y la depresión, y que, como ya les mencioné, por ende pueda llevarnos a problemas físicos: hipertensión, y de hipertensión enfermedades cardiovasculares, un sistema inmunológico debilitado por estos procesos inflamatorios que puede generarnos el cortisol. Entonces, una cosa nos va a llevar a la otra. Aquí abajo, como un resumen muy sencillo, el vínculo emocional entre el cuidador informal y el paciente puede ser muy fuerte. Puede tener hasta donde pueda alcanzar el paciente un apoyo, una gratitud, hacer esa conexión, pero conforme vaya degenerándose más el pacientito, pues obviamente va a haber menos satisfacción y va a estar más presente el sentimiento de culpa, porque los cuidadores creen que esto sucede a través de su responsabilidad y no tanto porque sea el eje que va a llevar la enfermedad como tal, ¿sí? Por lo que se proponen algunas actividades de intervención psicológica, que esto en algunos grupos que tenemos de ayuda, trabajamos para que se cumplan, o las personas que estamos captando dentro de nuestras investigaciones aquí en la región de los Altos es justamente que se tengan actividades de terapia cognitivo-conductual para ayudar a los cuidadores a que identifiquen esos patrones o pensamientos negativos y saber cómo dirigirlos y llevarlos, ¿no? Otra de las partes es el mindfulness y la reducción de estrés basada justamente en el mindfulness, donde se enseñan algunas técnicas para estar en el presente, en el aquí y ahora, y que el paciente pueda darse un respiro como tal, ¿no? Las habilidades de afrontamiento y el apoyo social y grupos de apoyo, que esto es lo más importante: saber de dónde está afianzado el cuidador para poder repartir o comisionar entre su mismo familia una actividad, y que él no se sienta una sola persona, se sienta con la responsabilidad de todo lo que gira en torno a su paciente: el cuidado, la alimentación, la medicación, las citas médicas, la rehabilitación, ¿sí? Porque, como ya lo mencioné, esto es algo desgastante, y es necesario que también pensemos no solamente en los enfermos de alguna enfermedad mental crónico-degenerativa, sino también en capacitar a los cuidadores primarios informales para que tengan el conocimiento de todo esto y puedan mejorar su calidad de vida y de paso la calidad de vida de sus pacientes enfermos. Muchísimas gracias por escucharme. Ha sido un placer compartir con ustedes esta pequeña presentación. Gracias al doctor Víctor. Y bueno, si tienen alguna pregunta, a sus órdenes.
Muchas gracias, Gaby, por la presentación. Sin duda es muy importante entender que además de la afectación que ocurre con el paciente, ocurre con el cuidador,
Así
¿no? Aún sabiendo que esas enfermedades son muy rápidas en su curso, el periodo en el que el cuidador se encuentra atendiendo a su paciente le genera bastante ansiedad, ¿no? Además de la situación que implica el ver el progreso de la enfermedad, bueno, más bien de los cuidados que lleva esa enfermedad, ver el progreso de la enfermedad, y creemos que en los casos que son sobre todo de tipo hereditario, puede existir todavía un poquito más de ansiedad. Ya nos comentaste sobre algunas estrategias en este sentido. Em, a- em, ¿Habrá algún estudio que nos diga cuál de todos estos que tú comentas ha presentado un poco más de eficacia o depende de caso por caso? No sé qué es lo que has
No,
encontrado.
sí, gracias, doctor. Más bien depende de no, no a todos les funciona lo mismo. Obviamente vamos a irnos por el que tiene más evidencia y los estudios han demostrado que la terapia cognitivo-conductual es una de las primordiales porque alcanza a captar al cuidador en el patrón de algo que pudiera estar haciendo mal y que está repercutiendo dentro de su salud. Las otras terapias de apoyo, por ejemplo, la de grupos de apoyo o la de red de apoyo también suelen ser buenas, pero la que tiene más evidencia científica sería la terapia cognitiva.
Muy bien, muchas gracias.
Gracias.
Francisco, ¿quieres comentar algo?
Agradecer. Gabriela, muchísimas gracias, la verdad, por tu participación en
Gracias.
este simposio. Es un placer el conocerte por este medio y esperemos en Dios que
Igualmente.
podamos seguir adelante
Que así sea.
en muchos más eventos como estos.
Que así sea, doctor. Muchísimas gracias por sus palabras y estoy a sus órdenes en lo que se pueda colaborar y seguir participando. Ha sido un honor para mí. Gracias.
Agradecemos, te agradecemos de corazón. Gracias.
Gracias. Hasta luego.
Muchas gracias. Bueno, vamos a avanzar en nuestra agenda. Vamos ahora con nuestro siguiente ponente, que es Nina Overbeck. Nina Overbeck creo que ya se encuentra con nosotros. Nina, are you here with us? Are you ready for?
I am. It's great to be here.
Thank you so much, Nina. It's always a pleasure to see you and mostly to hear you and to hear about and learn about all the advancements that you and your group have been doing for establishing a new drug or therapeutic approach to the prion patients. I will do a very quick introduction in Spanish, what I believe you will also understand.
Yeah.
So, let me just change the language for the audience. Bueno, se encuentra con nosotros Nina Overbeck. Nina Overbeck, como ya los comenté ya lo mencioné hace un momento en, en inglés, es una investigadora que se encuentra actualmente trabajando para establecer una nueva estrategia terapéutica para enfermedades por priones. Nina Orbeck es actualmente la directora del área se llama Preclinical Translation, ¿no? El área precisamente de este abordaje preclínico en Gate Bioscience, en donde se encuentra a cargo de los programas de esta empresa. Ella tiene un postdoctorado en Genentech y tiene un doctorado del laboratorio, del MRC Laboratorio para Biología Molecular de la Universidad de Cambridge. Es decir, un autor de varios trabajos de investigación en en muchas revistas, incluyendo la revista Nature. Y pues se encuentra con nosotros hablando sobre este tema. Ya ha sido expositor en varios congresos. La conocimos precisamente en el Congreso de la Fundación de CJD en Estados Unidos, y entonces hemos tenido una buena comunicación y siempre ha estado con toda la disponibilidad para apoyarnos y hablar con nosotros sobre los nuevos avances para un tratamiento de enfermedades por priones. Sin más, le dejo la voz a Nina. And Nina, welcome again.
Thank you very much for inviting me to speak. It's a real pleasure to share a bit about myself, about Gate Bioscience, and what we're doing to develop a treatment for prion disease. I'm sorry, my Spanish isn't quite good enough for me to give this presentation all in Spanish, but if I ever come to Guadalajara in person to present, I'll try my best and do it in Spanish next time. So as Victor mentioned, I am the VP of Translational Sciences at a biotech company called Gate Bioscience. We are a small molecule drug discovery company based in San Francisco in the Bay Area in the US. And we're a small company, currently 32 employees. And we've been working for the past 4 years to develop a pill to treat prion disease. And that's what I'll be talking about today. So hopefully from a previous talk from Eric and through previous knowledge, we all know that despite the different etiologies of prion disease and despite many major historical names for human prion disease, it's really the misfolding and aggregation of host-encoded prion protein, which is the causal agent for all prion disease. So when normal prion protein takes on this infectious scrapey form, this misfolded form, when it touches normal PRP, induces that to misfold, and you get a chain reaction that spreads throughout the whole brain, causing cell death and neurotoxicity and all the devastating symptoms of prion disease that we know. Now, when a disease is caused by a misfolded protein, traditional small molecule drugs and antibody drugs that bind directly to the pathogenic protein really struggle to eliminate that pathogenic protein. So these traditional modalities are really not the right tools for the job to stop aggregated proteins. What a better approach is if we can eliminate PRP at its source. So if we can prevent normal PRP from being made, then there's nothing for misfolded PRP to touch and to misfold. And this spreading and aggregation throughout the whole brain is halted in its tracks. And there are several companies already working on this approach. You may have heard of Sangamo Therapeutics. They have a gene therapy approach which silences PRP at the DNA level using gene therapy. Maybe even more well-known is Ionis Pharmaceuticals. They have an antisense oligonucleotide which targets PRP at the RNA level. They are currently in phase 1/2 clinical trials. And today I'm going to be talking about Gate Bio's approach. And we have a new small molecule that allows us to selectively prevent prion protein secretion. But all of these approaches result in the same outcome, which is that if you have no prion protein, then you have no misfolded prion protein, and therefore you are able to rescue the symptoms of this disease or delay or improve the symptoms of this disease. So first question to ask is, will eliminating normal PRP or PRPC work? And there's a lot of evidence to suggest that the answer to that is yes. Some of that comes from historical genetics that has been published. So we know that homozygous deletion of PRP in mice prevents prion infection. So you can inoculate these animals' brain with infectious prion protein and they will never develop the disease. So without PRP, patients should never develop prion disease. There's been a lot of published studies, including the example that I'm showing here. And this example is from therapeutic oligonucleotides developed by Ionis, showing that lowering PRP levels with ASOs can significantly extend the lifespan of prion-infected mice. And it's clear that the earlier you intervene, the better, but they still see a therapeutic benefit even after symptom onset. The next question to ask is, will eliminating PRPC be safe? If we all express abundant amounts of PRP in our brains, does it not have some kind of important function, and will eliminating it be problematic? And all the evidence points to the fact that lowering PRPC should be completely safe. Not all proteins have important functions postnatally. And there are actually many proteins that are expressed in our brains and throughout our bodies that are dispensable for healthy life. And we believe that PRPC is one of those, and there's a lot of evidence for that. There are actually humans that lack a copy of PRP, so they are heterozygous for the prion gene, and they are completely healthy. And there's been a variety of different knockout models made, including in goats and sheep. These animals seem to be completely healthy without PRP. Mice lacking PRP do have some kind of peripheral neuropathy as they age, but we would be able to monitor this in a clinical trial setting. So switching gears now, if we want to lower PRP in the brain to treat prion disease, how do we do this with the small molecules that Gate is developing? So Gate Bioscience, we are developing small molecules that we call molecular gates. And these are small molecules with a new mechanism to eliminate secreted pathogenic proteins. And it's important to lay out a bit of background. So in the cell produces many, many different proteins, in fact, tens of thousands of different proteins all the time. And these are the proteins that allow the cell to carry out its function. About a third of those proteins, about 4,200, are what are known as secreted and membrane proteins. So these are proteins that are made inside the cell, but their final destination is either outside of the cell or in the membrane. And prion is actually one of these secreted proteins. Now, when the cell makes these secreted proteins, they enter the endoplasmic reticulum on their journey from inside the cell to outside the cell. And they use a common channel to enter the endoplasmic reticulum. And this is a channel called Sec61, as depicted here. And all of these proteins go through this common channel, including prion. Our small molecules gate this entryway and selectively prevent prion from entering the secretory pathway. And what you get is essentially a selective elimination of prion protein whilst leaving everything else to be produced as normal. So because we can eliminate prion protein with a small molecule, this has a couple of key advantages that I want to highlight. The first is that small molecules, because they are small, they tend to be very brain penetrant, and they can journey to all of the regions of the brain. And we know prion disease is a disease that affects all regions of the brain. And a small molecule is just a better tool to be able to lower PRP throughout the whole brain. Small molecules are also orally bioavailable typically. And so this allows patients to potentially take this medicine orally or as a pill. So they could reach into their medicine cabinet and take 1 or 2 pills a day, for example. And this is very convenient for patients and their caregivers, as opposed to having to go to hospital to receive intrathecal injections, for example. So now I want to share a little bit of data with you all, and this is for a molecular gate tool compound that we call MG813. This molecule is quite selective for prion protein, and it's also very potent against mouse PRP. And so this makes a very good tool for us. And what you're looking at here is animals, mice, wild-type mice, dosed once per day with molecular gate MG813, either at 100 mg per kg for 8 days or 50 mg per kg for 14 days. And when you look at the amount of prion left over in the colon and in the brain at the end of the study, you can see we see almost complete elimination of colon PRP when the animals are dosed at 100 mg per kg, and about 90% elimination of colon PRP when the compound's dosed at 50 mg per kg. In the brain, we see very profound 80% reduction in brain PRP at 100 mg per kg and 50% brain reduction at 50 mg per kg. We wanted to take this compound forward into an efficacy model, and we did this in collaboration with the Broad Institute. And there is a very long prion efficacy model, which is well used in the field. It is very robust, but unfortunately, it takes a very long time. And how this model works is you take wild-type mice and you inoculate their brains with infectious prion or PRP scrapie, and they take about 120 days to develop symptom onset. And at this point, we treat these animals with 50 mg per kg once per day dosing of MG813. So this is really to mimic treatment at symptom onset, which would be what we would be doing in the clinic. And we treat these animals for close to 70 days. We ended the study at day 187. We hope that at the end of the study, all the animals in the vehicle group will have died and succumbed to the disease, whilst the animals in the control, in the compound treated group, would be surviving. And we looked at survival and body weights and also some measures of brain histopathology. So you can clearly see, looking at body weights first on the left, that as the study progresses, the vehicle-treated animals start losing weight pretty quickly and succumb to prion disease, whilst the animals treated with MG813 manage to maintain their body weight. On the right, you're looking at survival, and as we expected, when we ended the study, all of the animals in the vehicle-treated group had succumbed to disease, whilst we had most of the MG813 animals still surviving. And worth noting, of course, that dosing with this compound was well tolerated after almost 70 days of dosing. And we're working now to follow up with the Broad Institute to run a longer study to complete the Kaplan-Meier curve. What we need to do is formulate this compound into the food of the animals, because daily oral gavage is very stressful on the animals. So once that work is complete, we'll be starting the longer-term efficacy study. So that's it for data. I wanted to end just giving a small overview of small molecule drug development. Many people maybe aren't fully aware of all of the phases that a company goes through to make a drug, and I want to outline that. So it takes on average 13 to 15 years to go from an idea to an approved medicine. So it's a very long process, it's a very risky process, and it's also a very expensive process. And what you start with usually is what's known as the preclinical or discovery phase. This typically lasts 3 to 4 years. And at this point, you are making and testing thousands of different molecules and optimizing, making them more potent for prion, making them more selective for prion, and improving their pharmaceutical properties. Once you have identified the drug, the one that you know you're taking forward, you then take this medicine or this drug into what's known as IND-enabling studies. And this is essentially a rigorous set of toxicology tests in animals. Typically, this is done in rat and dog. And this is to ensure that when you first administer this drug to humans, it's going to be safe for humans. This typically lasts around a year. After that, we have 1 to 2 years in phase 1 clinical trials. And typically, this is done in healthy volunteers. Not always, but typically in healthy volunteers. And this is a first look at the safety and the pharmacokinetic properties of your molecule. If this looks good to go, then you can proceed to a much longer phase 2 or 3 clinical trial in patients. And the aim of this is to, of course, look at efficacy. Usually, approval of a drug requires a definitive clinical impact. This may be something like an impact on survival in the case of prion disease, but it can also be other clinical endpoints, such as symptoms, for example. And then if that looks good, then you can proceed to a drug approval, hopefully. So where is GATE on this journey? We are just finishing up our preclinical phase of our process, and we've identified our development candidate, and we're now proceeding into toxicology testing in animals. So with that, this is my last slide. I want to thank the Gate Bioscience team for all their amazing work on this project. In particular, Jennifer Pitson is the lead chemist on the project. She's the one that actually makes the medicines, which is the most important part. So I want to acknowledge her. And of course, our collaborators as well, Eric Minical, Sonia Valep, and their team at the Broad Institute. If you'd like to learn more about GATE and about our approach in prion disease, please take a look at a science article that was published last year called Foiling Deadly Prions. It has a really nice overview of the various therapeutics that are being tried in the space, and it does explain molecular gates and how our mechanism works. And thank you very much for your attention and for the invitation today.
Thank you so much, Nina, for this great presentation. I will ask, and I guess you mentioned, and I guess I will obviously see it or find it at the science paper, but last you are blocking the protein. So what is the fate of this blocked protein? Is it easily degraded? Does it accumulate, or I don't know?
Yeah, that's a great question, something I should have mentioned. So when we prevent prion protein from entering the ER and its translocation fails, it gets immediately ubiquitinated and degraded through the proteasome. So there are actually dedicated E3 ligases and dedicated mechanisms that ensure that proteins that fail translocation are quickly gotten rid of and never accumulate in the cell. So that's what happens to PRP.
Okay, great. And what will be the estimated population to be enrolled in the clinical trial? Are you thinking also on preclinical, only symptomatic?
Yeah, I think the initial idea is to go into symptomatic patients first. We don't have any specific details yet about the clinical trial, how many people, where the sites would be, but we hope to, as we are getting closer, to involve, the community as much as possible in that.
So maybe you can include Mexico.
I hope so.
Possible.
I hope so.
Okay. Thank you so much for this good and great presentation.
Thank you.
We will move on now in our agenda to our next speaker. And okay, so I guess we already have Dina with us. Dina, are you ready to?
I'm here. Hi, Victor.
Hi, I'm sorry. I guess I will just make a brief pause. I guess Francisco was waiting to say thanks to Nina. Is that okay, Francisco? ¿Le vas a comentar algo a
Yes,
Nina?
yes. I want to thank you to Nina. Es obrigado. Muchas gracias, Nina.
Muchas gracias and obrigado. Yes. Perdón, Francisco, se me pasó en esta ocasión.
No te preocupes. Nina, bienvenida.
Thanks, Francisco. Now we will go on with Dina. Dina, which is part of the CAD International Support Alliance. Maybe you can start uploading your presentation, Dina. So yeah, Dina. Yeah.
You see it, Victor?
I guess we can only see your screensaver from your laptop or computer.
It worked this morning when we tried, didn't it?
Yes, it did. We can see your we can see the presentation right now.
It's on my other screen, but I'll make it work.
Okay. In the meantime, I will introduce you Spanish. Dina Simpson es miembro de una familia donde se presenta la enfermedad de Creutzfeldt-Jakob de tipo genético. em, y derivado de ello, ella fundó la organización CJD Insight con el objetivo, pues, de ayudar a otras familias y que puedan abordar de mejor forma todo el trauma, el estrés y las dudas por las que pasan las familias en donde se encuentra el Creutzfeldt-Jakob de tipo familiar. Y a su vez es miembro de la junta directiva del CJD International Support Alliance. Em, ha sido promotora de manera muy activa de las actividades precisamente de esta red internacional para el apoyo de las enfermedades por Priones, junto con la codirectora, que es Susan Solvins. Ha participado en la presentación en varias ocasiones, perdón, en las internacionales, el más reciente que se realizó en Río de Janeiro, y a su vez ha sido de mucho apoyo para las actividades que hemos tenido acá en México como fundación, para la Fundación FendMex. Y sin más, pues le dejaré la voz a Dina para que nos hable un poco sobre la experiencia que ha tenido y sobre la forma en la que participa el CJD International Support Alliance. We're back with you, Dina, and I'll leave the microphone to you, and we're ready and happy to listen to you. Thanks.
All right, thank you, Victor, and thanks to you and Francisco for inviting us back to speak at this very important conference. We're honored to do so. I will apologize up front. I am struggling with bronchitis, so I'm hoping my voice holds out and my cough doesn't disrupt too much. Appreciate your patience and understanding. As I think Victor mentioned in his introduction, Suzanne Solvins and I are executive directors of the CJD International Support Alliance. Professor Richard Knight is also one of our executive directors. Suzanne sends her apologies. She is taking a well-deserved break and traveling after we were at the Prion 2025 conference in Brazil, but she does send her greetings and well wishes. A little bit of my background, I didn't understand everything that Victor said, but I picked up certain pieces of it. But I do come from a genetic prion disease family. We carry the E200K mutation for familial CJD. To date, we've lost 23 family members spanning several generations. Lost a cousin just almost a month ago on Sunday. So it's a journey that continues, especially for genetic families. But we do what we can. We remain hopeful. Our overall objective of the Alliance is really to represent and help represent the voices and faces of those that are personally impacted by prion disease. One way we do that is to attend conferences such as this and Prion 2025 and others to really share stories of those that are impacted. I think it brings the human side to the science side of everything that researchers and our pharmaceutical partners are working towards for treatment and cure. And we so appreciate that and want to just represent those that they're working so hard for. A little bit about the Alliance. We were established in 2006. We were at the U.S. conference, family conference. We were in the Washington Court Hotel lobby, as you can see in the picture. Suzanne really had the idea about bringing all of the support organizations together that existed to have a unified voice to advocate for and work towards supporting families and hopefully a treatment or cure one day. We had 4 support organizations present: Australia, US, Japan, and the UK. And we're really happy to share that as of November of this year, we do have now 19 member organizations that represent 18 countries. There's certainly more work to do. There's more countries that need support organizations because CJD and prion diseases don't discriminate. It's everywhere. It's a global issue, a global disease. And I'll talk a little bit later about how we're trying to help in that space. That's just a slide of all of our member organizations and their logos of their organizations, and we're grateful to have them as partners. And also at Prion 2025 in Brazil, we had 11 member organizations representing 10 countries attend the conference. This is the largest number of members I think we've had since we've been presenting at the Prion conferences. It was great to have the ability to meet them and see them in person. We come from different parts of the world with different time zones, and seeing them together and having that physical conversation and community was very special. Also, Ayrton, who is working to establish and formalize the organization in Brazil, actually coordinated a family meeting for Brazilian families to attend. It was very successful. We had speakers that talked about just the basics of prion disease, what it's like as a caregiver, the importance of genetic testing for those impacted by the genetic prion diseases. And we had all of our member organizations present talk about their work in their countries and how well their organizations are doing. And everyone's at a different stage, which is okay. It's a growth process. And so we learn from each other and we share best practices, which really makes us highly effective. We also had a couple of family members share their story, which is very important, as I talked about earlier. And it was very well received. And I think everyone enjoyed the time that they had to have that focus. We also had some of the pharmaceutical companies that are working on therapeutic treatments present. So having that time with families and our member organizations was very, I think, important and helpful to everyone there. I'm going to talk now about the three main objectives that we're focused on as the International Support Alliance. First and foremost, it's about assisting countries in establishing their support organizations in their native country, or for ones that are already established, expanding their service offerings. We feel this is very important because the regulations and laws in every country are very different. So when someone gives advice, maybe from a US perspective, that may not translate appropriately in their native country. So it's very important that these countries have their own support organizations so they get the best information, the quickest information that's going to help them navigate this journey. Second, we advocate for and foster for awareness around the impact and needs of patients and families across the care continuum, as well as the global community at large. And again, one of those is to really help bring awareness at conferences. And we'll talk about some of the other things that we're doing to help in that space. And lastly, to provide avenues for patients and families to partner with the pharmaceutical companies, the academia, medical professionals. I think, and we do that by leveraging support organization in countries that have a support organization. And those that don't, we have friends and advisors that help us, and we'll talk about that a little bit later. But especially as clinical trials become more real, it's important that we expand this community as much as we can, keep them engaged, keep them informed, so that when clinical trials do come up, if they want to participate, they're actively aware and they're ready to proceed. So it's going to benefit not only us, but these companies that are working so hard to find a treatment or cure. One way that we've been able, one program that we've established in the last 3 years is we have a grant program, and we use our funding to help our member organizations, as I talked about, establish support organizations in their country or expand services, as well as other things that we're using this for. So this is a slide just summarizing what we've done so far. In the last year, we've helped France and New Zealand have their first prion disease family meetings, where they bring their families together like we're doing here and share stories and hear from the experts in the field and the pharmaceutical companies. We've also helped New Zealand set up their helpline so that their families have that support. We've helped Japan develop a logo for their organization, and we've helped Portugal establish charity status, and we've secured funds to help India, Austria, Turkey, and Brazil to attain charity status. That obtaining charity status allows them to take in funding so that they can do more things and offer more services to their patients and families. So it's very critical that we can help in that space. We've also been able to award travel grants this year for the first time to our member organizations to attend Prion 2025 in Brazil. And I think that helped get more members to the conference so they can hear the most relevant and updated information. We have time together to talk about our needs and our goals and objectives and share information. And for the family meeting that I spoke about that Eriton coordinated, we were able to help provide funding for the AV support and the translation services that were needed. The funds also help us maintain our website so we have accurate information out there for our members and others that look for information, and then create our artifacts in preparation for CJD International Awareness Day every year that happens on November 12th. So this is a summary of what we've done in the last year. We're excited for more to come. And we really want to thank Ionis Pharmaceuticals. They have really helped fund our grant program the last 3 years. We appreciate their faith in us and their belief in the objectives that we have and their commitment to the broader prion disease community. We are so grateful to their support. On our website, you'll find we have a specific section for clinical trial updates for prion disease. Again, trying to keep everyone informed of what's happening in that space. You'll see on our website, we have the companies listed at the bottom. You can click on each one and find out where they're at, what updates they've provided us. So again, it's another way just to keep information at the forefront and accessible to those that are looking for it. Also like to take a moment, I mentioned our friends and advisors before, but really these are global support of experts in the prion disease field. They could be researchers, clinicians, or others working in the field. And we really rely on them, especially in countries that don't have support organizations, but also in their own native country. Some of the criteria for becoming a friend and advisor is they have to be dedicated to support patient associations in their own country and also to assist families in other countries where we don't have patient associations. And I know Victor has helped us in other countries, especially if they're Spanish speaking, and we appreciate his support, and others have stepped up as well. And we couldn't do what we do without them. They've been invaluable to us. And every presentation we give, we ask if you can help in your country, contact us. And I'm happy to say that after Prion 2025, we had during that meeting 5 to 6 experts come up to us and say, we want to help in our country and we want to help other countries. So we're expanding. I think right now we have 57 friends and advisors covering 27 countries. So we're very proud of that, and we look for continued growth in that space as well. And I'm just going to show a video of our friends and advisors. Again, we can't do our work without them, and they're great partners. We appreciate them so much. I'm just going to take the last few minutes to talk about International CJD Awareness Day, which happens on the 12th of November every year, and just some of the things that we've been able to provide. I think one thing this year we did was Remember in November. So this is just something that people could post on Facebook or LinkedIn or other social media platforms to help bring awareness. This really just is a word cloud of, the types of prion disease, the fact that research is happening, that we need a treatment or cure, and some of the characteristics that I think the prion disease community, patients and families bring to the table, their courage, their commitment, their faith, the community at large. it's just important to remember that part of it, what makes this community special. And we were able to share that this year. Last year, we also did a infographic on what the prion disease types are. We have expanded that. I wasn't able to include it in this presentation. I apologize, but it is on our website. It's a video that expands this into not just the types, but symptoms, common symptoms, diagnostic tools. It's a way of education and bringing awareness. This is helpful, I think, for clinicians who haven't experienced it before. It's just a quick summary. We'll make that into a larger PDF that can be downloaded and printed, and countries can translate it into their own language and use this as a tool to share. Just another way of leveraging information so people don't have to start from scratch.
Globally united, prion disease awareness. Tous dans le monde, sensibilisons aux maladies à vie. Globally united for prion disease awareness. We're globally united for prion disease awareness. Todos unidos para crear conciencia sobre las enfermedades priónicas. Prionios Emeraldaner Sekundarfinaro. Es wird vereint für das Verständnis von Prionenkrankheiten.
Globalmente unidos para sensibilizar por las enfermedades por prione.
Uniti in tutto il mondo per sensibilizzare sulle malattie da prion. Globally united, prion disease awareness. Unidos internacionalmente pela conscientização sobre doenças priónicas. Globalmente unidos pela sensibilização para as doenças priónicas. Wereldwijd verenigd om prionziekte onder de aandacht te brengen. Globally united for prion disease awareness. हम आगे सतह prion बीमारी की तहकीक के लिए मुतहद. Globally united for prion disease awareness. Weltweit vereint zum Bewusstsein von Prionerkrankung. Traendo y apuntar el derecho.
Globalmente unidos por la concientización sobre las enfermedades priónicas.
Prion måste identify communities united issues.
So that was a video that we created to of all of our member organizations to really put out the message that we're globally united, that this is a global disease. And that was created last year. We added our new member Turkey to it this year, and we continue to use that as CJD Awareness Day, just again show the impact worldwide and how we're globally united and really want to stay connected and work towards a treatment or cure. So that's what I had to share with you today. I think, again, I appreciate the opportunity to share what we're doing and how we're trying to, work across the globe and keep people get the help they need and the support that they need. So thank you, Victor.
Thanks, Nina, for this presentation. And I know that in the past years, you as a group, the CJD International Sports Alliance, have also spoken at the international prion meetings. What do you think will be the message that the researchers are getting from the foundations? I mean, are they realizing that there's more to do than just focus on the technical issues? Do you think the message that you as a foundation is being transmitted to them? I know you're like a bridge in between the families and the researchers. Do you think this goal is being accomplished by being there at the prion meetings?
Absolutely, I do. I think, again, telling what we do at the conferences is whoever the host country is, we engage the host organization if there is one, the sport organization. If not, we look and find families that are willing to share their story. So we had 2 families share their story in Brazil, and we've done this at every conference that we've attended. And the feedback that we get is very powerful. I think, again, it just brings that human story. It brings the human purpose to their work. I think they've said they find it very motivating. And I think that's important. I know we appreciate everything that they're doing, the researchers and the pharmaceutical companies. And I think it helps them to really, I think, motivate them, light a fire maybe, and just to know that their work is important to so many people, that they continue to do what they're doing, because we need them. And we need the newer generation to get engaged and get interested in this field of prion disease, because it's not going away. So we need these folks to commit, and we're grateful to that community who is committed. The pharmaceutical companies, I think, have been equally open to really working with us, and I know they've worked with Debbie to really talk about running their ideas by us about how the clinical trial looks and what does it mean to patients and families, what barriers might they have. So they're very engaged and they're very committed. And I think the more we tell the story and the more that we can collaborate together, it's going to be a really good outcome. We're very hopeful.
Great. It is good that the doors are open both ways, from the research to the families and from the families to the researchers. And
and I think even between researchers, to see a research community come together and share, not say I can't, they share because they know this is a rare disease. It's fatal. And the only way we're going to find a treatment or cure is if we all work together. And that is what's happening.
Yes. And hopefully we will have that, those new drugs soon. We already had a couple of approaches from Eric and Nina, and we believe that also by spreading the voice through the researchers, the clinicians, and the foundations, and the international organizations such as the one that you are leading, will be also very good for promoting these efforts and for having the families to know that this is happening and everybody's working together for that. Thank you so much, Tina, for this talk, and we will have to move on the agenda with our next speaker. And I don't know if Debbie, I guess, was already.
I am here. Yes, I am.
Thank you so much, Debbie. Thank you so much for joining us and for spreading the voice from your side, from the CJD Foundation. I will introduce you in Spanish and then move back to you, okay?
Okay.
Okay, bueno, tenemos con nosotros y nos da mucho gusto, es un gran honor para nosotros también tener entre nosotros como ponente a Debbie Jobs. Debbie Jobs es actualmente la presidenta de la Fundación para Enfermedades por Priones de Estados Unidos. Como pueden ustedes ver ahí, ya es la CJD Foundation, y organiza cada año la reunión que se llama CJD Foundation. Tienen el número de cada año. La más reciente se realizó en Chicago, y con su grupo de trabajo también forma un puente muy importante entre las familias y los investigadores y las demás organizaciones que se encargan para el estudio de las enfermedades por priones. Después de ella hablará el doctor Brian Apoli, que es el encargado del Centro de Enfermedades por Priones de Estados Unidos, y juntos forman una dupla, consideramos que muy buena, una muy buena organización de trabajo que llevan ellos para apoyar con las estrategias de diagnóstico y las nuevas estrategias o enfoques de tratamiento para estas enfermedades. A su vez, han sido un gran apoyo para las demás organizaciones a nivel internacional. A México nos ha apoyado bastante, y desde México le mandamos un gran agradecimiento a la Fundación y al Centro para Control de Enfermedades por Priones de Estados Unidos, porque han sido también pues promotores de las actividades hacia México y de México hacia Estados Unidos. Nos han permitido establecer en contacto con las familias. Y bueno, sin más, le dejo la voz a Debbie Job para que nos hable un poquito de estas actividades y que llevan a cabo en la Fundación de Estados Unidos, la CJD Foundation. Thanks again, Dina. I'm sorry, thanks again, Debbie. And I will leave the microphone to you to hear about the wonderful job you're doing as the president of the CAD Foundation in the US.
Thank you, Victor. And are you seeing my screen okay? Good.
Yeah, we can
Okay.
see your screen and we can hear you very well.
Good, good. Well, thank you again for the invitation to speak and to share a little about our work with you. This is a screenshot of our most of our team at our annual conference. Let's see. Lost my mouse here. Okay. So just going to give you a quick overview of our various programs at the CJD Foundation. Most of our work, as you'd imagine, starts with our helpline. We have calls daily, 7 days a week from families, as well as from professionals and public and sometimes the media. Families understandably often ask about diagnosis, caregiving questions, genetic testing, how to get tested and what the results might point to. The general public often concerned about transmission risk. We have questions about treatments and clinical trials and much more. Fortunately, because of our partnership with the National Prion Disease Pathology Surveillance Center, we are able to refer families to a free autopsy program, an MRI study, and a very neat study that Eric mentioned earlier, the Teleneurology Assessment Program that Dr. Brian Appleby administers. We are sometimes able to point families to an expert or a physician, genetic counselors, and various studies they can participate in. And of course, they're looking for resources. So we've got a lot of online materials. We have recordings from our conference sessions, recorded webinars, caregiving binders, and more. And we also invite them to engage in our support groups, our annual conference, and we introduce them one-on-one to caregivers. We offer 40 to 50 support group sessions a year, and often that starts with the current caregivers group and unfortunately moves on to inviting families to participate in the bereavement group. We also offer groups for genetic families and drop-in groups for both the Eastern and Western time zone. Our website is sort of like another employee for us that works 24 hours, and lots of people tell us that's the first place that they interacted with us. We've got lots and lots of recordings of sessions and webinars, information about caregiving, frequently asked questions, and much more. One of our missions that's very important, and we realize more and more how important, is helping facilitate medical education. So again, partnering with Dr. Appleby and others, we try to arrange as many medical lectures as we can each year. And those can be in person, they can be remote, and they can cover a variety of groups and issues. We've also exhibited at the AAN Neurology Conference as a great way to let neurologies know about the impact of the disease on families and about the availability of our services for families. And of course, when we're contacted by medical providers, we provide referrals and educational materials. We're very proud of our research grant program and very grateful for the families who tirelessly fundraise to fund these research grants. We've awarded over 90 grants and almost $6 million throughout the history of the program. This year alone, we awarded $850,000 in grants. The grants can cover anything from basic science, treatments, and more. And we've started offering fellowships, which meets another of our missions, which is to inspire early career scientists to join the field and stay in the field. Each year we do go to Washington, D.C. for an advocacy day. And of course, we conduct advocacy campaigns throughout the year. We're very pleased that this year our advocacy led to something called report language going into the appropriations bills. So not just a request for funding, but also an explanation by members of Congress of why it's needed. Again, our family members are very gracious in donating their time to come to DC and participate. And we've gotten great turnout and great results. We also recently managed to persuade members of the House and Senate to support a resolution to recognize CJD Awareness Day. And that's important because it's great for families to see and decision makers to see these members of Congress speaking out about the importance of our work and public health experts' work. I mentioned our annual conference, and Victor, we're glad that we've had you with us at that conference on many occasions. This is a great opportunity for both families and professionals to hear each other's views and experiences. So naturally, we do have lectures and keynotes. We have family support groups and workshops, lots and lots of Q&A sessions. And I think one of the very favorite sessions is the moving roundtables, where families can sit in small groups with experts and get a lot of their questions asked. I was one of the participants in this before I worked at the foundation, having lost my husband to a genetic form, and that was always my favorite part of conference. So you are very welcome to join us. Next year's conference will be July 10th through 12th in Chicago. Part of the fuel behind all of these programs is our Strides for CJD campaign. So each October, at about 20 locations around the country, groups of families host these events. And they are excellent fundraisers that support our research and other programs. But they're also sort of like a walking support group for those of us affected. It's a great way for us to pay tribute to our family members who we have lost and connect with our community and other affected families. So that is the end of my talk. And if you have any questions, I'm happy to respond.
Thank you so much, Debbie, for the presentation. It is indeed a great amount of work that you do in the whole country. And we know that moving the meeting from the usual place, which was in DC to Chicago, also helped to have some families that couldn't attend the annual meeting to actually be there. And do you think that this will help if you move the meeting to the western part, because flying from the west to the east in the US is not that easy, and that may also help to have more families to attend the meeting.
Yes, it's a very good question. We have done mini conferences before on the West Coast, but the West Coast is a pretty expensive place to host. So we will be considering that with our board and deciding.
Yeah.
But you're right that making it at a drivable location in the Midwest definitely upped the number of attendees.
Yeah, we saw that in Chicago this year, how the number of participants increased, which is good, because I believe that a lot of families know that the CJD Foundation is there. They do want to be in contact with you, with the researchers, and but they just can't attend for any reason. And having this mobile meeting through the country, I guess, will help a lot to make the family.
It does. Yeah, it does. And we've also been offering small travel grants for families who are first-time attendees to help them attend. And of course, we always record the sessions and post them on our website.
Yes, yes. Sometimes Francisco, the president of the Mexican Foundation, sends us the link of the presentation, so we also get to see the presentations that you have throughout the year. And I guess this is also a very, very good idea, because you're spreading the buzz every day. Every day, there's always someone listening, and there's always the foundation attending the needs of the families.
That's right. It's unfortunate to say, but when we hosted the conference in July, there were plenty of people who had not yet ever heard of CJD who now are very unfortunately interested because of their loved one's illness. So there are always new families coming who need that recorded information.
Yes. I don't know, Francisco, if you have something to tell to.
Yeah, David, thank you very much for being with us. It's a pleasure always to have you here.
Thank you for inviting me, Francisco. It's my pleasure as well.
And also your support with you is very important for Mexico.
Absolutely. We're glad to have anyone with us who's able to join each year.
Thank you very much.
Thank you.
Thank you so much. So we will have to move on now to our next speaker. I don't know if Brian is already there. I guess he's been around for some time during this symposium. Is Dr. Appleby? Yes, he's there. Well, we are also very, very happy and very welcome to have with us Dr. Brian Appleby. Dr. Brian Appleby has been attending families and cases with prion diseases for a long time. He's a psychiatrist, and is now at Case Western Reserve University at the CJD National Surveillance Center. He's the head of the National Prion Disease Pathology Surveillance Center, and he collaborates with the CJD Foundation and with a lot of organizations in the world. And we're also very, very, very happy to have you, Dr. Appleby, with us. And we're ready to listen what the National Prion Disease Pathology Surveillance does, which is a lot. And we will like to have an idea of all of this tremendous amount of work that you and your staff does for the diagnosis of prion diseases in the country, and also helping some other countries to have some diagnosis. So, Brian, we're ready to listen to your presentation.
Great. Can you hear me okay? And you see the slides?
We can hear you okay, and we can see the presentation very well.
Awesome. Thank you so much. Victor, thank you so much for the invitation. I think I did this last year and I really enjoyed it. I've been on and off all day, but from what I've seen has been really exciting and top notch. I'm so happy that you're able to do this for your families and also your local healthcare providers. So today I'll talk mainly about the National Prion Disease Pathology Surveillance Center and also some of its activities. So first, I'll just provide a brief overview of how we do prion disease surveillance in humans in the U.S., and then I'll focus a little bit more on the activities of the Prion Center itself. And hopefully I'll be able to convince you that prion surveillance is really important. And then we'll provide some basic info on our findings as well. And I apologize, I'm in the middle of a migraine, so I only see half the presentation that you do. So hopefully this goes okay. So prion disease surveillance in the US, we have three primary methods that we do this. So the first is autopsy confirmation, and that's done at the Prion Center. We're funded by CDC to do that. And I'll talk about later why that autopsy confirmation is so important. The second thing that we do is we count RT-QuIC positive cases. You heard about that, I believe, from Dr. Zaire. That really very specific test for prion disease that's so specific that basically you can consider it a positive case in the right scenario. So we're able to count those. And then the third way we do is in the US, we have death certificates on any person who dies. And basically, it's their identifying information as well as their cause of death. And the Centers for Disease Control has access to that database, and they're able to search for all the variety of diagnoses that could indicate that that person passed from a human prion disease. So this is just a graph that kind of shows how we use all three of those methods together to do prion disease surveillance. So the yellow are cases that the only indication that we have that that patient had prion disease was because they went to autopsy. And then the NCHS is the National Center for Health Statistics. This is that database I was referring to that basically has the death records for why someone passed away. And what CDC is able to do is look for cases that were diagnosed with prion disease and then match it with our autopsy results and our spinal fluid testing. So that's what the gray and the orange are. Now, the blue is basically just death certificate data. So someone passed away from CJD on their death certificate, but we don't have any other data. So what you'll see here, if we look at 2015, is that the vast majority of cases somehow get confirmed as having prion disease through the Prion Center, through either the autopsy program or spinal fluid testing. What's also important is, unfortunately, we do see some cases that have prion disease mentioned on their death certificate, but on which the autopsy was not suggestive of prion disease. So we're able to remove those cases from the numbers for that year. Now, if we look at the incidence of prion disease, the incidence is described as the number of new cases that happen every year due to a particular disease per million individuals. So across all ages, we have about 1 to 2 new cases per million individuals every year that pass away from prion disease. And that's pretty true across all the countries. The harder you look, the closer you get to 2 in a million, but largely most countries are in that range. However, not everyone gets prion disease. It tends to be a disease of mid to late life. So if you separate out the incidence by age, you'll see that older people over the age of 65, have a higher incidence rate. And that incidence rate is about 4 to 7 people per million individuals will develop prion disease. And on the other hand, younger people are much less likely to get prion disease. And I know we talked a little bit about this before, probably with Dr. Knight, who unfortunately I missed, and Dr. Geschwin, who only caught a portion of. But most people don't think in public health statistics, which is what the incidence rate is. So another way to think about this is the number of deaths that happen every year due to a disease. And if we were going to do that for prion disease, it's about 1 in 6,000 deaths every year due to prion disease. That also means that everyone has a lifetime risk of about 1 in 6,000 chance of developing prion disease. So again, that 1 in a million number is very helpful for public health, but I don't think it's super helpful when we talk to family members or even clinicians sometimes. So RT-QuIC, as Dr. Zaire mentioned, has really revolutionized the field in terms of diagnoses. We're able to make a probable diagnosis of CJD with a positive RT-QuIC in any neuropsychiatric symptom. So that's very, very powerful ability to diagnose a disease. And because of that, we're able to actually capture cases that may be prion disease just by spinal fluid testing alone, which we weren't able to do before with diagnostics before we were doing RT-QuIC. So it's greatly improved clinical care and clinical diagnosis, but it's also greatly improved our ability to do public health statistics for prion disease. So what this graph demonstrates is the green is just the number of autopsies. So this would be the incident rate. So it's 0.85 per million individuals per year. And then the blue is what happens if you take the autopsies and match it with people who didn't go to autopsy but had a positive RT-QuIC. As you can see here, depending on the year, it almost doubled our ability to capture cases of prion disease in the country just by looking at RT-QuIC in people that didn't go to autopsies. And this becomes important later on when we start talking about what we've been seeing in more recent years. So RT-QuIC, very important for diagnosis, but very important for surveillance as well. So the Prion Center has a variety of missions. It was started in 1997 at Case Western University, mainly in response to the variant CJD. That's the kind of CJD due to eating meat contaminated with mad cow disease, but also medically transmitted or iatrogenic CJD. And it's funded by the Centers for Disease Control and Prevention. And it was started originally under Dr. Pierluigi Gambetti. And we're tasked specifically with doing neuropathologic surveillance for the CDC because brain tissue analysis is very important for this illness. It's the only way to definitively diagnose the disease, but very importantly, it's the only way to know what type of prion disease it is. So we can determine whether or not it's due to variant CJD or with genetic testing, whether it's genetic, or with history, whether it's through medical transmission or whether it's sporadic. And it importantly allows us to also look for new forms of the disease. So we've been doing this since 1997. So we have a large collection of brains and we know what sporadic CJD looks like. So by looking at cases that come through year to year, if we see neuropathology that looks different, that could indicate to us that maybe we're seeing a new prion disease. So as you can imagine, our focus is mainly on preventable and potentially new forms of prion disease, like variant CJD, medically transmitted CJD, or some new disease. And unfortunately, in the US, the thing that we're most concerned about is something called chronic wasting disease. This is a prion disease in the cervid species. So that includes things like deer, elk, moose, and caribou. Think about the four-legged friends that have antlers. Those are basically cervids. And this is the map. Fortunately, it's not up to date because of government situations in the US. But as of April, you see that it's affecting 36 of our states and also affects Canada. It also somewhat affects some Scandinavian countries, but really North America, with the exception of Mexico, is by far the most heavily affected by chronic wasting disease. The concerning thing about chronic wasting disease is it's very easily transmissible amongst its own species. And that's partly because prions are excreted in their saliva and their urine and their feces. And they're also free-ranging animals. So they're not like cattle where they're domesticated and they're penned. And if someone was positive or some animal was positive, we could eliminate that whole herd. These are just animals that just go about their day and they don't really care what state they're in, what county they're in. They just travel with no ability for us to really track them. So, we really don't see an end in sight for chronic wasting disease in animals. We only are expecting it to continue to increase in prevalence. But importantly, we don't know whether or not this prion disease can transmit to humans. Hunting and venison consumption is extremely common in the US. And our main fear is that potentially chronic wasting disease could transmit potentially to humans, either through eating those animals or another animal eating a chronic wasting disease infected deer and then it being transmitted to us. So that's really our main purpose at the Prion Center now is trying to determine whether or not this is happening. To date, we don't have any evidence that this has happened, but as you've heard with prior stories, the incubation period for these diseases is measured sometimes in decades. So this could already be going on and it could be transmitted to humans and we not know it yet. So that's why continued surveillance is so important, especially in the US. So I'm just going to review some of the aspects of the Prion Center. Our main focus, of course, is that neuropathologic surveillance that the CDC funds us to do. And we do autopsy coordination, coordinating autopsies around the country of cases with suspected prion disease. And as you heard before, we characterize these cases to make sure that they meet expectations for what we know about current human prion diseases. In order to help our autopsy program, we try to help clinicians make the diagnosis. So we also have a clinical laboratory that offers spinal fluid testing, 1433 total tau, which you heard Dr. Zara talk about, as well as RT-QuIC. And we also offer genetic testing. A lot of what we do is research, particularly about characterizing cases of prion disease so that we know what we're looking at in case we see something new, we can identify that it's new. We also are continually trying to improve our diagnostics. And of course, we have a large biobank of brains and spinal fluid that we use for our research, but also share with the international community at large for their research as well. We do a variety of outreach and education, presentations, publications. We also have a website, and a lot of the outreach is through the medical education project that Debbie talked about through the CJD Foundation. We consult with a variety of stakeholders, physicians, state public health departments, and then we have two programs that I'm going to talk about in detail today, and that's the MRI consultation program and then the teleneurology assessment program for CJD, which apparently we've already heard about earlier today that I missed. So hopefully I won't be redundant in that. And then finally, very importantly, we're in an age of experimental treatments. So I think it's also our responsibility to help guide and inform clinical studies and to help provide quick access to diagnosis as well as knowledge of potential clinical trials that people might want to participate in. And of course, we ourselves also participate in clinical trials. So now we're going to present data of the Prion Center itself. So this is a graph, at least for the last 25 years, of brains that have come through the Prion Center. So the top line, the blue line, is the number of referrals or the number of brains that we get every year. And then the orange line are the number of brains that wind up being prion disease. So as you can see, not every brain that we get winds up being prion disease, only about 30 to 40%. And the ones that aren't prion disease are a variety of things, mostly other progressive brain diseases like Alzheimer's disease, but sometimes other things. As you would expect from what you've heard earlier, the vast majority of these cases are sporadic, meaning they just happen spontaneously. They don't have a known cause. And in our country, about 10 to 15% are due to a genetic mutation of the prion protein gene. At the bottom here, thankfully, you can't see the blips because these are the acquired prion diseases. As you heard from Dr. Geschwind, in the US, we've had 4 cases of variant CJD, all of which developed the disease or acquired the disease, I should say, from outside the United States. And then we've had 15 cases of medically transmitted CJD, most of which were from human growth hormone that were taken from cadavers. So this chart shows CSF samples or spinal fluid samples that we received that was RT-QuIC positive. And as you can see, that's steadily been increasing over the years, especially last year. And I'll talk a little bit about why I think that's the case. But if you remember that earlier graph that I showed you about the power of also including RT-QuIC and when you capture cases, that's what this graph shows for the most recent years. So your blue line is just RT-QuIC positive cases. So the last slide that I show. And then the red line is what happens when you combine the people that go to autopsy that were prion positive to those that had a positive RT-QuIC that didn't go to autopsy. So this is what we would call definite cases and probable cases, all determined by laboratory analyses. And you'll see here there's a couple spikes, and I think some of them are fairly easily explainable. So this spike in 2019 is almost certainly due to the change in the CDC's diagnostic criteria that says that you can have a positive RT-QuIC with a neuropsychiatric syndrome, and that meets the definition for probable CJD. But also, that is the year that because of the diagnostic criteria change, we started running RT-Quick on all of our spinal fluid samples. What we were doing earlier is what actually Dr. Zer determined that we probably shouldn't be doing, and that was we were screening cases by total tau. So now we run RT-Quick on everything. And then you'll see 2014 was a spike. We don't have a real clear answer to this. There's nothing different on our autopsy rates. It seems to be largely driven by RT-Quick positive results that didn't go to autopsy. But the one thing that happened in this time frame is the rollout of that Ionis clinical trial using antisense oligonucleotides. So we think that the possibility of having a clinical trial for a disease may have actually increased the testing and the actual finding of cases of prion disease over that year. Again, we look for patterns of cases that we know exist. There are a variety of different subtypes of sporadic forms of prion disease. That's what all these gobbledygook acronyms mean down here. But the take-home point here is that if you look at the distribution of all the different types of sporadic CJD, you'll see that it really hasn't changed over time. And this is important because we don't know what chronic wasting disease would look like in a human. So we need to make sure that it's not mimicking another form of sporadic CJD and that we're misinterpreting it to be truly sporadic when it's a new prion disease. And we would do that by looking for different changes in frequencies of the different subtypes over time. And we've not seen that. We do the same thing with age. For reasons that aren't entirely clear, acquired prion diseases tend to occur in younger people. So when variant CJD, it occurred in people in 20s, 30s, or teens, 20s, 30s. And that's unusual. Same thing for medically transmitted CJD. But as you can see over time, and also by subtype, that the mean age of onset of disease is pretty stable. And in fact, it's actually gone up over time, which is what we would expect because the US has an aging population. So that's comforting to see. These are a list of all our different mutations that we've seen in the US through the Prion Center. Our most common mutation, which is also the most common mutation around the world, is E200K, followed by B210I, as well as the mutation that causes fatal familial insomnia. I did want to point out this mutation. This is the T188R mutation. I think that this is probably overrepresented in Mexico because we do have a lot of these cases. We have 29, and almost all of them are Hispanic or Latino descent, and a lot of them have family members from Mexico. So we think that this is a very common mutation in Mexico. What's important to know is that it seems to be a pretty highly penetrant mutation, meaning that if you carry that mutation, you're very likely to get the disease, but it doesn't seem to be 100%, meaning that it's not guaranteed that you'll get the disease. So I don't know if Victor has any information on this mutation in Mexico, but this is something that we would be interested in learning more about in your country. I'm going to talk a little bit about some of our programs that we have. One of the programs that we have is we'll look at MRIs for families and clinicians from around the country and let them know if we think that they're suggestive of CJD. That helps them with the diagnosis, but it also helps us alert them as to the presence of our brain autopsy program, but also potential clinical trials that they might be interested in. So in roughly the last year, we've had 76 referrals, and most of them wind up being suggestive of CJD. And the ones that aren't, that went to autopsy, wind up being a variety of different unusual subtypes. And what's interesting is we do receive MRIs from foreign countries as well. We don't have funding for that, but we will do that. I believe the neuroradiologist who reads them for us in Italy, Dr. Bizzi, reads them for free. So this program is very important because we've got a lot of important research out of it. So one was Dr. Bizzi came up with a new criteria for detecting prion disease on MRI. Now, you heard about two criteria already through Dr. Geschwin's talk, the European criteria and then the UCSF criteria. And this is also slightly different as well. Instead of looking for two regions of areas that could be suggestive of prion disease, it only includes one. I'd like to say that these criteria are probably more similar than they are different. Although this criteria allows us to capture more cases, I think we have to be careful with it because, again, it's just one spot in the brain that they're counting. So it really should be used with caution in a proper clinical context and by a neuroradiologist who has seen a lot of these cases. We're also able to subtype sporadic CJDs. You heard about this a little bit from Dr. Geschwind as well. So we're able to create an algorithm that's able to, with pretty strong accuracy, predict the subtype of sporadic CJD, and that's helpful, especially when you're thinking about things like clinical trials or even just prognosticating for family members, because different subtypes have different illness courses. And of course, you can't know the subtype until the person passes. That's not helpful for them. But if you can predict it with a reasonable amount of accuracy through the MRI, then that might be able to help the family out. And then we can look at cases that have had multiple MRIs over time that have gone to autopsy and maybe predict where the disease starts. So this was a study that did that, and we were able to determine that depending on the subtype, the prion propagation process started in certain areas in some subtypes, in certain areas in other subtypes. And that's important for things like drug development, in which a drug might not reach certain areas, but may reach other areas, and that some people may be more likely to respond to one treatment, but not another because of where the disease starts. The other program that I'll talk about briefly is the Teleneurology Assessment Program. This is a study that is completely done remotely over Zoom, just like we're doing today. It allows us to follow people over time. We originally did it to try to collect as much natural history as possible for surveillance. More recently, we've looked at caregiver well-being and burden and really treat it as more of a natural history study to collect data to help people properly design clinical trials. We like to call this the little study that could, because it is an extremely small study. It's two people. It's me and a research coordinator and a very, very tight budget because the only expenses we really have is salary support for our time to have the time to do it. And I don't know if Bob Ross, the famous painter, is popular in Mexico, but he's very popular in the US. And one of his famous phrases is happy little accidents. So he would be painting a painting and then he would slip and he would say, that's okay. It's a happy little accident. We'll make that a happy little tree. And in many ways, this study is a happy little accident. We did not design it to do what it's being used for, but we're happy that it is being used in this way. And of course, now that we know it's being used to help design clinical trials, we've tried to actually reflect the protocol and that it would help that. So we collect something called the MRC Prion Disease Rating Scale, which is a rating scale that looks at a patient's functional ability specific to prion disease. This was developed in the UK by Simon Mead and colleagues. And then we also look at cognitive function as well, looking at a test called the TICS. And we're able to determine a lot of things. So first off, the reason why this is so important for clinical trials is how frequently we're seeing patients. Before, when people were doing clinical research in person, you only really saw a patient who was symptomatic maybe once or twice because the disease course was so severe and they couldn't travel back to the researcher. But remotely, we can see people throughout the entirety of their illness in the comfort of their own home or in their own hospital or hospice. So, and as you would see, we're seeing people almost every month, and that mimics what you would do for a clinical trial. So these data are very important for that. And you can look at these data and say, hey, it might be important to look at the different trajectories depending on their score. And you'll see here around the 14 to 16 point mark is you see that these people have a different trajectory compared to people down here. So that maybe you would want to focus early clinical trials on people that are around that 14 to 16 or above level. And we did a study using these data with the help of Ionis to also look at, well, what would be the meaningful within patient change that could determine whether or not we're actually seeing an effect of a therapeutic agent. And it looks like 1 to 3 points might actually be enough to say that this therapeutic compound is actually making a significant difference in this patient's illness course. And then the other thing that we looked at is caregiver burden. This is the first time I presented these data, so hopefully Debbie's watching. Yeah, partly why the CJD Foundation wanted to fund this project is to look at caregiver burden. We don't have a lot of people in this study because we've only been doing this for a year and a half or 2 years, looking at caregivers specifically. So we only have about 30 people. But we did have some preliminary findings. One is that if you look at neuropsychiatric inventory, these are neuropsychiatric symptoms, you'll see that over time they tend to get worse and then they peak and then they get better. And if you look at caregiver distress over time due to psychiatric symptoms, that's the blue line, you'll see that that actually gets much better once the neuropsychiatric symptoms start getting better, which is good to know. But if you look at the overall caregiver burden of the family member, which would be the yellow line, something called the Zaret Burden Interview, an overall measure of how burdensome the illness is, you'll see that that's pretty stable over time, regardless of the neuropsychiatric symptoms that they're seeing. So that's important to know that burden happens throughout the whole course of the disease. And actually, it's really high. These scores are comparable to other really burdensome illnesses, for example, frontotemporal dementia, much more burdensome than what we typically see in Alzheimer's disease and Parkinson's disease. So what we can say is neuropsychiatric symptoms peak and then they improve, but caregiver burden remains stable, likely due to other factors. And the other thing that we found, again, Zaret Burden Interviews, that overall marker of caregiver burden, is that the caregiver tended to be more burdened the younger the patient was. So if the caregiver was caring for a younger one, they tended to have more caregiver burden than an older patient. So again, these are just very descriptive analyses, but I think it's a good start, and it lets caregivers know that we hear them and that we're studying them and we know how best to help them. So I'm just going to end up with some challenges that we do have in the US. Last year, another lab started performing RT Quick. Now, that could be great because we have more resources to do RT Quick, but as you heard from a surveillance standpoint, we use RT Quick to count cases. So if another lab is doing it, we may miss cases. This lab actually has been very open with us, and they do want to share their data with us. It hasn't happened yet, but I do think it will happen. And if that does happen, then I think we don't have to worry about losing cases. We do have decreasing autopsy rates, and that this is a national trend for all illnesses and across the world, so it's nothing new. But we also worry that because RT Quick is so specific for the disease, that maybe people don't feel the need to do an autopsy. And that may be true. You may feel confident that it is prion disease, but importantly, RT Quick doesn't tell you anything about what type of prion disease it is, and it's certainly not going to tell you whether or not it's a new prion disease. And then, of course, we have issues with funding. We are very thankful for the CJD Foundation, who keeps pulling us back from the brink of being defunded. But this is a constant challenge. And of course, as you saw from that map with chronic wasting disease, this is probably the worst possible time to end funding in the United States. So we're thankful for their support, not just for our families and for our programs, but also for keeping us in business. So with that, these are some of the wonderful people that we work with. We have a great group of people that really care a lot. Some of us have been with us for a very long time. I want to thank Victor again for inviting us to talk about our work and just for really an excellent symposium. So thank you for your time.
Thank you so much, Brian, for this great presentation and for letting the people in Mexico know the great amount of work that you're doing as a surveillance center in the US. As for the T1 mutation, we do have seen some patients in Mexico, and from what I've heard from one of these families, there seems to be a cluster of families in some in a place in Mexico. We've tried to be there and try to study this family because this is a very small city, kind of isolated. So we believe that the degree of endogamy is very high. maybe adding up to the number of the cases in Mexico. The problem is that this small city is in a hotspot in the country. So to get there is kind of dangerous, but we've tried to be there because we've heard about this center at least from two different sources that they say, well, we know that there are a lot of cases in there. So maybe some of this increased number of these genetic cases in Mexico that you found also in the center may come from this part of the country. So we are hoping to get there in a safe way, so we can do the studies in there. So we'll let you know about this. And with this past family, Eric helped us to with our small of funding to do the sequencing. We already found that in at least in three generations we found positive cases. So we're starting to do the genetic counseling on these family members, which is also a question that I had for the to you and the surveillance. How did you work with the families and the clinicians for the genetic counseling? I know the genetic cases for every disease is very problematic for the clinicians and also for the families. So how do you manage to do this with clinicians and families in terms of the genetic counseling?
Yeah, so again, I missed a lot of Eric's talk, so I don't know if he talked about this. I apologize if I'm repeating it. We just do the clinical lab testing, right? So it's on the ordering provider to make sure the patient gets genetic counseling, right? We don't know whether they do or not. We strongly suggest it. Anyone who I see personally in clinic definitely has to have genetic counseling. But a lot of people want to get genetic testing, but they don't want it in their healthcare records for a variety of reasons, right? So in collaboration with Eric and also Debbie and the foundation, we hope to start a study shortly that allows us to do it remotely and anonymously. In that case, we will know that everyone gets genetic counseling, everyone gets access to their genetic results, but it's anonymous as well. So did Eric talk about that at all?
No. Okay.
So I expect that will change in the next year or two how we do genetic counseling in the US.
And are you doing next generation sequencing for the study of the genetic mutations?
Yes, we do that, and we do and Sanger sequencing as well.
Okay. Uh, Another question that we hear a lot from the families is let me say if I can say it properly. What is the average turnaround time of a diagnostic for the Arctic week, for the genetics, or for the immunohistochemistry?
So our average turnaround time for spinal fluid is about 5 business days. So we've drastically increased that. Our turnaround time for genetics on blood is about 2 to 3 weeks. Our turnaround time for autopsy brain tissue is about 3 months.
Oh, okay. Yeah, because when you have cases in Mexico, and as you know, we don't have a formal lab for doing that in Mexico. So usually a lot of the samples are sent to the surveillance. So they almost all the time ask for that. Okay. And about CWD. And as we've all seen that it's been moving from a very low spot in the US to being widespread in the US. And there's been some counties in Texas that are right next to the border in Mexico. So I will say that I'm pretty sure that one of those tiers are already in Mexico. And unfortunately, there is not that surveillance to know that in our country. There is a surveillance going on for cattle in Mexico, but I haven't heard of any CWD case in Mexico. But I believe that it will soon be in our country, and it is also a great work that you're doing, because that's one of the big questions. Is there going to be a case for CWD as it is for BSC? We know that variant CJD can be acquired from the BSC cases. I know that the surveillance has done a very exhaustive job looking for cases that may have CJD through CWD, and you up to this moment you have found no correlation. But it is also good to see that you are looking for atypical cases, which is also very good in terms of surveillance. Do you think that in some of these brains that you haven't found CJD, which are known as maybe Alzheimer's or any other atypical case of dementia, do you think there could be one case in there, or maybe at least work as a control group to contrast the data with some other atypical CJD cases?
Are you asking, am I concerned that the prion negative cases could be a chronic wasting disease case?
Or any other type of atypical CJD cases?
Yeah, and not so much, because, when you do brain analyses for prion disease, we do a variety of different tests, as you know, right? We do a Western blot, we do immunohistochemistry, something called H&E. So we really have a lot of different ways to determine if something is prion disease or not. So if chronic wasting disease goes to humans and it doesn't really look like prion disease at all, and none of the prion markers are positive, I would think that would be extremely unusual. So I would expect that some of the prion tests would come back positive, but they would look unusual, kind of like how variant CJD looked unusual. So like you said, it's very important to have those positive controls of what we know exists and is supposed to exist versus what may be new and indicative of a transmission of a new disease.
Well, thanks. These are all of my questions so far. I know there are a lot of questions to ask to the surveillance, but again, Brian, I'm very thankful to you and all of the surveillance, not only for this presentation, but for the great job that you do for diagnosing cases in Latin America. I know you receive a lot of samples from Latin America. And the current idea and goal of the foundation in Mexico is to finally get the lab working in Mexico. So hopefully we'll be there soon, and maybe cut the number of samples a little bit from the samples that you receive in the US. But so far, and I will say forever, we're very, very grateful to you for all the help that you do to the clinicians and the families in Mexico and Latin America. Thanks so much.
Thank you, Victor. And exciting news that you guys are going to get your own RT Quick lab. That's awesome.
Hopefully it will be there pretty soon. Well, we need to move on in the agenda, and I guess we're coming to the end of the meeting. We have Francisco Motat, and Francisco Motat is part of the Mexican Foundation in FendMex. He's the son of the architect Francisco Mota. So sometimes when we're at the meetings, I say Francisco Jr. or but I'm very happy to have him in the meeting. He's been in the symposium for the whole time, but I guess it is a good moment to introduce him. He's also been around. He was in the meeting in Chicago this year, so he's been introducing into the works of the foundation of the Priones, and he will be in charge of closing the symposium, of giving the closing words of the symposium. Así que, Francisco, te dejo la voz, que qué bueno que has estado aquí en todo este rato y escuchando las ponencias, y pues te dejamos el micrófono para que puedas dar las palabras finales y hablar también un poco de lo que hace la fundación aquí en México.
Sí, muchas gracias por la introducción, Víctor. Como mencionas aquí, hemos estado todo el día al pie del evento. First of all, good afternoon to everyone. As we come to the end of this incredible symposium, I would like to take a moment to express my deepest gratitude. First, to each of our speakers, thank you. Thank you for your generosity in sharing not only your knowledge, but also your time, your research, your experience, and of course, your heart. Your work gives family around the world something that is priceless, hope. So thank you very much. Muy buenas tardes a todos. A todas las personas conectadas hoy, familias, cuidadores, médicos, investigadores, estudiantes, organizaciones, muchas gracias por haber estado aquí, por haberse conectado. Su presencia nos recuerda por qué hacemos esto y para quién hacemos esto. Durante estas horas hemos aprendido muchas cosas sobre historia, diagnóstico, avances en el tratamiento, el papel del cuidador en estas enfermedades, la labor de las fundaciones en el mundo y sobre todo el propósito que nos une. Y nos une algo muy poderoso, la convicción de que ninguna familia debe de recorrer este camino solo. Desde México, a través de FendMex, estamos trabajando para crear apoyo, conciencia, colaboración científica alrededor del mundo, como lo hemos vivido el día de hoy en el evento. Nuestros proyectos incluyen actualmente, como lo hemos platicado, la construcción del primer laboratorio especializado en diagnóstico e investigación de enfermedades por priones en México, programas de apoyo psicoemocional y, por supuesto, un pilar que tenemos en la fundación son estas alianzas internacionales con todas las fundaciones y organismos que están a favor y apoyando estas enfermedades. Somos una fundación joven, pero profundamente comprometida. Y hoy, por supuesto, quiero invitarlos a que se sumen a este movimiento y que nos puedan apoyar de diversas formas, como es colaborando en investigación y divulgación, aportando experiencia o guía, compartiendo esta red con las personas que lo necesitan y, por supuesto, participando en los proyectos y eventos que FendMex tendrá próximamente. En esta causa hay espacio para todos. Creemos firmemente que el futuro del diagnóstico, la investigación y el acompañamiento de enfermedades priónicas debe construirse juntos, entre países, entre instituciones y entre familias. Las enfermedades priónicas avanzan rápido, pero juntos podemos avanzar más rápido todavía. Juntos aprendemos, juntos nos apoyamos, juntos nos damos esperanza. Gracias a cada uno de ustedes por su humanidad, su ciencia, su participación y su corazón. Desde México, con respeto y mucho cariño, gracias por ser parte de FendMex este año. Gracias por ser parte del evento.
Muchísimas gracias, Francisco, por este cierre. Y creo que con esto concluimos, ¿verdad?, las actividades de este tercer simposio de enfermedades por priones. Estamos muy agradecidos con todos, con las organizaciones en nuestro país, obviamente con FendMex, y por todos los ponentes, los investigadores, los integrantes de asociaciones que estuvieron aquí con nosotros. We're very grateful to you all. I know I can see that Debbie, Dina, Inga are still here connected. We thank you all for your help and your support. We hope to have you in person very soon, hopefully next year, if we can make it. Well, hopefully in the following years. But we will be very, very honored to have you again with us in this symposium as we've had in the past years. Muchísimas gracias y pues cerramos. Y esperemos que haya sido una sesión de mucho provecho para todos, para los investigadores, los integrantes de la fundación y también para las familias y personal de la salud. Creo que estoy ya nada más secundando el mensaje que dijo Francisco. Yo no quiero ser muy repetitivo. Por mi parte, pues yo también me despido de ustedes. Muchas gracias y que pasen muy buen día y muy buen fin de semana. También.